யூடியூப் பாருங்க. யூடியூப் பாருங்க.
Photo network coverage. Yes, sir. UP.
Okay.
The demo is also.
I don't know.
Even the best for us,
அதமா இது தெரியலையா?
தெரியல சார்.
தெரியல சார்.
தெரியலையா? இந்த ரொம்ப தான் முன்னாடி பார்த்து தெரியுமா? ஐஸ்கிரீம் வந்துச்சுல? வந்துச்சா? சவுண்ட் வரும், சவுண்ட் ஆகுறீங்க.
Hi.
Good evening. I can.
How are you?
Fine. Very good. Very nice. After long time.
யாரும் வரலையா?
வருவாங்க. இன்னும் டைம் இருக்குல்ல. 6:35 ஆகுது. சரி 5:35 இன்னும் 6 o'clock தானே. So they will join around 5:50.
சென்னையில இருக்கிறியா?
ஆமா சென்னை தான் வேற எங்க எல்லாம் சென்னை தான். Within 4 walls. Very rarely go out. Of course, time spending on the webinar talks, listening, and also presenting a lot of time.
சரி.
இன்னைக்கு கூட ஒரு 100 webinars attended. 100 webinars attended in the 4 months. அதான் டைம். அப்புறமா நீங்க சென்னை தான் இருக்கீங்க. மைக்கேல் சென்னையா நாகர்கோவில்?
நான் சென்னை தான் இருக்கிறேன். லாட் ஆஃப் வொர்க்ஸ் டு டு, சோ அதுலயே டைட் ஆயிடுச்சு. கரெக்ட் பண்ணிட்டு தான் இடையில திடீர்னு மெஸ் எனக்கு ரிமைண்டர் வந்து ஒண்ணு ஜாயின் பண்ணேன். அப்புறம் கால் எப்படி இருக்கு? கால் எப்படி இருக்குன்னு
அப்படியா? சரி கேட்கிறேன்.
கால் எப்படி இருக்கியா?
கால் நல்லா இருக்கு. லெக் நல்லா
ஆ.
ஒன்றும் அது எப்போ திடீர்னு ஒரு வீக்ல ரெக்கமெண்ட் சரியா போயிட்டு கொஞ்சம் ஸ்ட்ரெயின் பண்ணி இப்பதான் வீட்லயே இருக்கும் இல்லையா, எக்ஸசைஸ் பண்ணி வாக்கிங் மட்டும் போவேன். மற்றெல்லாம் எல்லாம் ஆன்லைன் பர்ச்சேஸ் எல்லாம். இங்க தலைவர் ரொம்ப வரல, தலைவர் இன்னும் வந்துருவாரு.
அவருக்கு கட் டைம்ல வருவார்.
இன்னும் வந்துருவாரு. இன்னைக்கு வந்து ரொம்ப சீக்கிரமா பார்ப்போம், செக் பண்ணி பார்த்து. வெரி நைஸ். வேற எங்க இதுக்கெல்லாம் தரம் போறீங்களா சென்டருக்கு யூனிவர்சிட்டி?
இல்லையா, போய் 7 மாசம் 8 மாசம் ஆச்சு. யாரும் வரதில்ல. ஃபேக்கல்டி மெம்பர்ஸ் மட்டும் வர்றாங்க, ஸ்டூடெண்ட்ஸ் யாரும் கிடையாது. ஹாஸ்டல் திறக்கல. அது கேட்டாங்கன்னா யூஸ் டு சஜஸ்ட். நம்ம அங்கெல்லாம் போறதில்ல, எங்கேயுமே போறதில்ல. Just morning ஒரு 5 minutes நாயை கூட்டால ஒரு 500 meter போகும் அவ்வளவுதான். Otherwise, I walk about 10 kilometers within my house. Up and down.
Up and down.
Garden அப்படின்னு சொல்லி average about 12 to 14,000 steps and 9 to 10 kilometers.
Good exercise.
அதான் பண்றது.
இது நாகர்கோவில்ல போனீங்களா? இல்ல இதுவரை போல.
இல்ல எங்கேயும் போகல. 6 o'clock தானே? Then I will join back.
ஆமா நான் join
back.
Okay.
एक 2 का होगा, एक 3 का है, एक 4 का है, एक ऐसा कॉर्नर वाला है, ऐसे है, लेकिन वो बहुत छोटा-छोटा है।
Dr. Gupta, good evening. You have to turn on your mic, your microphone.
Good evening.
Good evening.
Good evening, everybody. Good evening. This is Dr. Gupta here.
Yes, well, nice seeing you. Nice seeing you here.
Yeah.
Right, sir. Right. Oh, Thomas Artung is awake now. Bye.
Hello.
Thomas, good morning. Can you hear me? Yeah. So early in the morning, I didn't want to disturb you, so I put you at 10 o'clock in the morning. Very nice of you to have joined. Nice.
Dr. Chandishanath, good evening.
This call is now being recorded.
So good morning. Sorry, I was my mouse didn't work. Oh, my trackpad.
Nice, sir. Nice, very nice to see you, sir.
No problem at all. Appreciate the invitation. Thanks a lot.
Thank you, sir. Thank you so much.
Good evening, everybody.
Good evening.
And if you are able to hear me, please raise your hand, and then we know that we are audible to each other. Now, before I say welcome, I think I will give the phrase, what is called as a phrase of this year, 2020, are 3 phrases. One is mute yourself. That means if you are not speaking, please mute yourself. The second phrase is unmute yourself. When you want to speak, please don't forget to unmute yourself. And third common phrase is you are not audible. That means you have to check it. And the fourth is please stick to time. And that's why we start dot at 6 p.m. And these are the few instructions. So I, as the president of this society, welcome you all. And the last phrase is, please declare that you are alone in your chamber and there is no children, there is no animal to disturb your conversation. So these are the few important announcements and phrases of the meeting. So please ensure that before you continue to be in this meeting. I, as a president of this prestigious new society, welcome all the delegates from India and abroad. And I must say that while welcoming you all, I feel sense of pride because this is only 3rd meeting, annual meeting, and it has grown in this 3 years in such a tangible manner, in such a productive manner, and it's such an important policy direction giving body. We never imagined so. And the entire credit of this, I will not say entire, but the major credit of this goes to Dr. M.A. Akbar Shah, who conceived this idea when we met 2-3 times. And then another important person is Dr. Raisuddin and of course Dr. Vijay and then the other team. But I would say the person behind the entire thing is Dr. M.A. Akbar Shah's spirit and enthusiasm. That is why the entire team today, which you can see with in this meeting, is there. And he is the person who has done the knitting of the experts of global in here. And I can see the former president of Japanese Society of Alternate to Animal Experimentation. I can see the representation from different countries. From Dr. Thomas is here, Dr. Michael is also here, and the top most people in animal experimentation, and particularly those who have the regulatory interest. I can see Dr. Chandishwar Nath is here, Dr. Krishna Sharma is here, and also Dr. Pant and Dr. Parmar from Indian Institute of Toxicology Research, and the only institution which gives so much emphasis on toxicology, Hamdard University. I will just give you also a background of this. We met a couple of times and we thought that we have been talking about the 3R principle, which is now more advanced version. We call it as a 4R, sometimes we call it as a 5R, but basically that based on the 3R principle. We have been talking a lot, but why not we should create a forum where we should develop some policy documents, and we also promote the development of animal alternative in India. How to generate support for that, and not important is just to discuss about it. And it is also important to propagate that, and also important is to convince the regulator to adopt them. And we have been able to, with some success, at least 2-3 parameters which have been able to convince them. It is also important that we gather information, expertise from the people from outside India, and then incorporate that in Indian scenario. We have a couple of institutions like IITR, Indian Institute of Toxicology Research, the Hamdard University, Central Drug Research Institute, then the Trivandrum Institute, and they all are contributing to this. And that is what important thing is. And I personally like to thank Dr. Akbar Shah, Dr. Rais, and Pratiksha, who is a young, dynamic lady from Bombay. And of course, Dr. Vijay, who is our dynamic vice president. And I also thank the society, I also thank the National Center for Alternate to Animal Experimentation, Bharati Dasam University, where our Dr. Akbar Shah has started the crusade of alternate to animal experimentation. I have a few things to say. That our objective is not to act as an activist against the experimentation on the larger animals. Never. Because I have been supporting that also. Because that has a important bearing in regulatory toxicology itself. But the important thing is, wherever possible, wherever feasible, we must work towards the equally evidence providing alternate to animals and go on reducing lower and lower animals. In India, zebrafish is now, at least 60 centers are now zebrafish. People are working on that. And working on toxicology, working on efficacy. And it is important that these centers get strengthened, supported, new methods about neurotoxicity, developmental toxicology, cardiac toxicity, and chemicals toxicology is percolated. And the idea is not to only promote the established scientists' work, but the idea is that they promote the lower level or middle level institution to adopt these things, what we call as catch them young. We are all, as a pharmacologist and toxicologist, Chandishwanath is Dr. Srinath and Dr. Pant, Dr. Parmar are here. At one time, when we were working on an animal for vomiting, for dogs, or maybe for cardiovascular system, and the animal experimentation or larger animals were stopped, we were most frustrated lot. But now we realize that it was perhaps not required to do all those things. If you think rationally, we could have done for that information maybe with the lesser number of animals or with alternative animals. So I would urge, because we have a very good mix of the policymakers, we have a very good mix of those who are representing the animal welfare committees, and for that matter, I also always say that I am also as much as animal lover as PETA or any other these organizations. But I also support a balanced approach. A balanced approach means you should not say radical treatment for both. You develop the animal experimentation alternatively more and more and more, support them scientifically, support them mutual benefit, mutual cooperation, give them funding and give them incentive, and we must generate evidence so that the larger animal experimentation are replaced. That's the important thing. And that can be done when you have. So at the same time, when we advocate, in fact, I advocated that there is no need of animal experimentation in undergraduate MBBS. And I was the chairman of Indian Council, the Medical Council of India. That we gave some logic and it was agreed and it was lauded. And so when we say for drug development, I think we have to be cautious because you have to change the regulatory process also. So we are a very rational society and we work for alternate to animals. We work for international collaboration. We work for collaborative approach between academic institutions, scientific laboratories, CDRI, IITR, Indian Institute of Science, Jamia Hamdard, and Thiruvananthapuram Institute, and the regulator. We should never forget involving regulator into our discussion, because ultimately what we are doing is for regulator or for education policymakers. So with these words, I again welcome you all, and the COVID-19 has taught us two important things. One is, if you have a will, you can do things. And you can do things in an alternative method. Now, when there was a conference, which used to be physical conference, now this is an alternate method. So you have developed an alternate method, which is, if not, I would say it is more effective. You start dot and time, you finish dot and time, and attendance is, you don't go for sightseeing during the conference time. So attendance is 90% or 100% there. So alternate system has worked better. We never tried it before. So we never knew the potential of this. Similarly, we should learn that alternate method to animals we should try. We should generate evidence, and I am sure that this will be more effective. So with these words, I again welcome everybody here, and I congratulate the entire organizing team, particularly of Dr. Akbar Shah and entire his team, Dr. Raisuddin, Dr. Vijay, and Pratiksha, who did extremely. See, time we used to work for a physical meeting for 3 to 7 to 9 months preparation. Now this has been done in shortest possible time. The gestation time is now reduced to, I think, 1/4 with better products. So I think we have done it, and we welcome you all. Thank you very much. And in Hindi we call as Namaskar, which is a now popular international way of greeting. And thanks to Corona also. Thank you very much. Namaskar. Over to Dr. Akbar Shah.
Rather, over to Dr. Raisuddin to chair the technical session. Dr. Raisuddin.
Dr. Raisuddin, are you there? Or you have to unmute yourself, perhaps?
Dr. Raisuddin, have you joined? Or you have to unmute rather, as Dr. Gupta said?
Dr. Raisuddin, I have seen Dr. Raisuddin.
You've seen it.
Doctor. We're unable to hear you, Dr. Raisuddin. I can see him speaking, but not able to hear him.
When did you see him speaking, Dr. Raisuddin? Dr. Raisuddin. Can you see Dr. Raisuddin? Can anybody see?
Hi, Dr. Gupta. This is Dr. Deepti from PETA India. I can see Dr. Raisuddin.
Yes, sir. I can see.
Then you may also be looking at whether he's mute or unmute.
He's unmuted.
Then I think he has to cut off.
He's not on mute, sir. Yeah, we can't see him though, but yeah, he's not on mute.
Then he has to log. Dr. Raisuddin, you have to log in because there is some problem in your mic.
Or
audio system.
A headphone.
Or headphone. Yeah, you remove your headphone and then see?
I think he just left the meeting. He might be rejoining, sir.
I think so.
He just left, so he might be rejoining.
So I would also mention one thing by the time he is joining, that the theme of non-animal method for vaccine testing is so crucial.
Can now anybody hear me? Hello?
Yes, Dr. Raisuddin.
Yeah, that's great.
Can you hear me now?
Yes.
Okay, thank you. So sorry for this disturbance. And as Dr. Gupta already has introduced the subject matter of the conference and also our small life cycle of 3 years, and we are thankful that everybody has joined. And from Jamia Hamdard, the University Delhi, as well as on my behalf, I welcome everybody. So in this session, which is the first session, plenary session, we have 3 speakers. And just a few hours back, rather around 4 o'clock, Dr. Parmar, Devin Parmar, who was about to speak on the new assay systems of peripheral blood expression profile to monitor exposure to environmental chemicals. He has some medical emergency at his close relative's place, so he may not be speaking. So, but we would have loved to hear from him. So we directly proceed with the first speaker of the day, who is Professor Hajime Kojima. He has joined us. Hajime?
Yeah.
Professor Hajime? Okay.
Hello? Can you hear me?
Hello.
Yeah.
Hajime. Yeah. Welcome, sir. And
thank you very much. Yeah.
It was very good evening there in Japan.
Yeah.
So we are very delighted to see you here. And Professor Hajime will be speaking on establishment of Asian Consortium for 3 hours with Society for Alternative Animal Experiment, India. So he will be rather speaking about the efforts which Japanese society has been making and has been pioneering, doing the pioneering job in this field. And I would just briefly introduce Professor Hajime. He is a PhD and is also Secretary General of Japanese Center for Validation of Alternative Methods and the Section Chief of Division of Risk Assessment, Center for Biological Safety and Research at National Institute of Health Sciences, Japan. He holds several publications. He is contributing a lot in this alternative field. And in every conference in India he has attended, he came to Delhi and he also came to Mumbai, and by his own resources and contributing greatly, encouraging us. He is also vice chair, which is working on the national coordinators of test guidelines program, and is also an expert in skin and eye irritation testing alternative method. He is also associated with OECD, so his contribution is immense. Without elaborating further, I would like to request Professor Hajime Kojima to please begin with his first plenary talk of this international conference. Thank you very much. Please.
Yeah, thank you very much. So the introduction means, can you hear me? It's okay? And also to share the presentation file in the establishment of the Asian Consortium of the 3R with SAAEI. Are you okay? Okay. Okay. Thank you. Hello, everyone. So good evening and Thomas, good afternoon, good morning. Sorry. Okay, I'm very happy to meet you again. So thank you very much inviting me on third national conference of the Society of Alternative Animal Experiment in India. I appreciate the president of Dr. Gupta and also to Akbasha. So thank you very much for support to the invitation. And so Dr. Lazin is to introduce me that I'm to working with the National Institute of Health Science in Japan and also former president of JSAAE. So on behalf of JSAAE, I'll talk about the Asian consortium, Asian collaboration with the whole 3 years. So I have no conflict of the COI. Okay, so today's agenda is firstly I'll talk about JSA activities, and also next one is to explain the Asian Congress. And so I think it is important is the international communication with Asian countries. Finally, talk about the bylaw of the Asian alternative animal experiment. Okay, so do you know so this is a website of the JSAAs. I think the purpose of the promotion of 3 years, replacement, reduction, refinement. So it's important is to promote and propagate of 3 years in Japan in research, development, and education. So our research groups established to this this society to 1982. And so reformed to Science Association 1919. And so we organized the World Congress in Tokyo 2007 and organized Asian Congress, first Asian Congress is coordinated in Japan 2016. And so this year we held coordinated 33 times annual meeting this year. You know, the Dr. Sakai, president of the JSAE, will organize the 33rd annual meeting of JSAE this November 22 and 12 and 13. So symposiums, one is for COVID-19, related to COVID-19, so foreign of the antiviral drug vaccine development. Second symposium is in vitro skin sensitization. And so third symposium, International Exchange Committee Symposium, International Trend of the COVID-19 Research and Skin Sensitization Testing. So Dr. Akbar has talked about this symposium. he talked about to update of the COVID-19 research in India. So I think that you enjoy the webinar is also to this our societies. Unfortunately, so this year's only webinars regarding the COVID-19. But so next year we are planning with the 34th annual meeting with JSAE is in Okinawa. This meeting will be held in the to November 4 to 6 in Okinawa next year. I hope to you we will to join the and meet you in the Okinawa next year. So anyway, so Dr. Sakai is here joining this meeting. So he is to hope to set up the cooperation of Asian counterpart community. Our effort and contribution concerning 3 years are very much appreciated in the international committees through the hosting World Congress 6 in Tokyo and First Asian Congress in Karatsu. While putting the basic of these efforts, we need to consider our new strategy for ideally benefiting the both Japanese, Korean, and world committees. So, Dr. Akbasha to participate in this Congress, to Asian Congress 2016. So, 4 years ago, so we coordinated the first Congress. I coordinated the co-chair of this Congress. I hope that you will enjoy this Congress. Anyway, and so purpose of this Congress was first conference of in kind of researchers from Asia and afforded an opportunity for promoting alternative method to research in Japan, whereas the concept of the 3 R's is just now archivic penetration. The Asian Congress is extended to archive multi-missions, which including disseminating information not just on the latest advances, including pure scientists, but on practical application of the 3 R's worldwide. And so next is 2 years later. So Chinese Korea coordinated the second Asian Congress in the Ganzhou in China. So this meeting is very successful meeting in China. So Dr. Pan, he is also to work in Institute of Disease Control Prevention in the academic military medical science. Dr. Pan is coordinated this meeting on behalf of toxic testing, alternative and translational toxicology in China. So almost 800 people joined this Congress. So we enjoyed the second Asian Congress. So 2 years ago. So and also to JSA, I hope to it's important international communication in Asia. So we communicate with KSA, Korean Society Alternative to Testing, since 2006, and also to sign the memorandum with Indian societies last years. So you remember? We signed the memorandum of understanding in Dr. Sakai and Dr. Gupta, and also to very successful ceremony in the last years. So I appreciated your support to the memorandum ceremony, and thank you very much to say that to instead of Dr. Sakai. Anyway, so I think that to JSA is to memorandum is to ST, European Society Toxicology in vitro, and European Society Alternative Experiment with also to and ASCCT, American Society's Computer Cellular Toxicity. We signed the memorandum with all societies, and also we collaborated with KSAE and India Society, and also in the future we hope to sign the memorandums of Chinese societies. So for more 10 years, JSA has continued to promote the mutual interaction with the annual meeting. In the future, we are planning to collaborate with the Chinese Society or TATT and TTAM to encourage activities related to alternative to animal testing with ASEAN Consortium. So what means ASEAN Consortium? Means so we have to start a discussion of the sort of Korean Corey and so Chinese Corys. So I wish to adapt it to next August. Name of the organization this shall be the Asian Society of Alternative Animal Experiment. The official abbreviation of this name shall be Asian AAE. The purpose of the society shall be to foster scientific cooperation among countries and promote progress in alternative animal experiments in the region. In practical, the supporting the Asian Congress of AAE. Okay. So I think that it's important is also to membership countries, to membership societies support the Asian AAE, and so we selected the council members and also to executive committees support to this to all members. So in the future, we have to worldwide societies, so to including Thailand and also Malaysia and also Taiwan. So I think that the key members to establish that to Asian AAE. So my worldwide proposal. The third Congress is now being planned for Korea during the 2022, and to promote close collaboration between our Asian colleagues. We are also now exploring the possibility of organizing for 2021 Asian AAE, which will provide the funding and the human resource in support of the future Congress in ancient countries. I wish SAE, and so Indian colleagues will join the Asian AAE and approve the formally fourth Congress in 2025. I wish to Asian colleagues to do international collaboration for 3 years worldwide. Okay, thank you very much. Thank you for listening. I wish you health and safety. Thank you so much.
Thank you, Professor Hajime. Thank you, Professor Kojima.
Yeah,
so since there is one speaker is missing in this session, so if Professor Phage Gupta and Professor Akbar Shah allow, we can have some brief discussion because he is proposing very important aspect of Asian society. So if any member like to comment, then we can invite the next speaker, because one speaker, 15-minute slot we have free. If 3-4 minutes we can discuss this very important issue which Professor Kojima has brought to this forum. And I think we can also invite comments from Thomas and other speakers and who are attending this program. Professor Gupta, please. Sir.
No, I agree with you. I think we can have 3-4 minutes quick discussion, and let us have any question. If anybody has a comment, then we can put it.
My question is that initially we are viewing only 3 countries like Japan, South Korea, and India. So can we expand? Can we find out some other societies if they are in the Asian region, especially in China, Hong Kong, or maybe other places, including if we expand our this horizon to even Australia, like that. If that's my suggestion.
Yes. Thank you very much. It's a good question. So in the future, we have to collaborate with Australia. But the first is we collaborate with Thailand and Taiwan.
Yeah.
So we have to cross countries to China and India. So also the Philippines and also to Malaysia is very important countries.
I have just one point. I think excellent presentation. do you think that what should be the scope of collaboration of our ultimate objective in India, what we feel is to develop some alternate method, at least target 1, 2, or 3, and get them incorporated into our regulatory system. So my point is, instead of having there can be two approaches. One is having a wider canvas of developing alternate to large number of things. The other approach could be identify 2 or 3 priority areas, low-hanging fruit, and work extensively on that, generate evidence, and get them incorporated into regulatory system. So what you think, what should be the right approach so that there is an impact is visible?
Yeah. Very good. Thank you very much. Good question. Yeah, I think that Japan, so China, and so Korea and India is also very good society. But so to the other countries that are not no society also 3 years and alternative. So in the future we have to collaborate in to establish that the societies of each countries, and so to also to education and research and encourage the research and education and the training of 3 years, and so also we will support to establish to encourage the society in each country, for example, developmental countries. So I wish to support to international collaboration, communication. Thank you.
Professor Hajime is bringing that initially he wanted to support strengthening of the societies either from the scratch or those who are little bit existing, to support them first to generate kind of starting material, and later on as Professor Y.K. Gupta is suggesting, ultimately we have to incorporate in the legislation and regulations, then only we can see the tangible outcome of our efforts. Yeah.
So I will just start the discussion with to law. Is that to based on our law is to discuss to communication. Yeah. Anyway, so we have to establish the rule of the ASEAN communication.
Professor Bangla is from industry side. If you would like to comment something, Subramaniam Bangala ji, Mani. We can't hear you. Unmute kar de. Just we can be unmuted. He can be unmuted if
Dr. Akbar wants to say something.
Thank you, Dr. Hajime, for your wonderful proposition. We have been discussing this matter for several years, starting 2016 at the Asian Congress that were held in Karachi. And I would like to tell you that the Indian Society has resolved in the last meeting held in Mumbai to associate with any attempt to make a federation, and therefore we have already resolved to join the federation. So it's a decision, and you can take it that we are there in the federation. And so this is the declaration. I take the president to confidence and the other members of the society to confidence to tell you that we have a resolution to the effect that we are joining the federation, number 1. And number 2, I have a suggestion in that the name of the organization which you are proposing be not a society, because we are already societies like Indian
Confederation.
Society. Confederation, consortium of the Asian Societies for Alternatives to Animal Experiments. That would be a better approach, because if you say a society, Asian society, then it would like dual membership. Member of the Asian Society, member of the Japanese Society, member of Indian Society. That wouldn't be nice. Better we have each one have their own national level society, and let these societies form into federation or consortium.
Confederation and union. Union may be another.
Federation. The federation, consortium, anything is fine. Yeah.
Union is also a good word, I think. All union like that. So you need to think the nomenclature, I think. Anybody else?
I am only giving a suggestion. I mean, you can put across to the other colleagues in the other member countries, societies, and take a decision on this matter. And my comment for the third suggestion that in 2025, India would host the Asian Congress. We will definitely put it before the general body and take a decision on this matter. But pending decision, I would like to tell you, taking again the consent of the chairman, the president of the society, that we are positively oriented towards a recommendation to that effect. But then I can't declare it that we are going to do it. I will have to take. I mean, the office bearers will have to take the approval of the general body for this decision. But likely we are going to have it in 2025 in India. Thank you so much for this suggestion.
So if I add to what Dr. Akbar Shah has said, I would say that keeping the societies, individuals, countries, societies name intact, I think the common name of federation consortium is important. So that should be considered strongly. That is one. The second thing is, we declare today our intention to hold 2025 meeting. With that we declare. What decision we will take after the general body, but intention we fully declare it.
In principle, agreed.
Thank you, very good suggestion. Thank you.
Mani, would like to add anything?
I have seen Dr. Michael's hand. Please, Dr. Michael. Dr. Raisuddin, to you.
Yes,
thank you, sir.
Let
Michael.
Yeah.
It's a good initiative, and I have only one clarification. You have the federation, all members of the individual association will become automatically members of the federation, or they have to enroll themselves individually as members. This point has to be clarified. Because many of the federations and other things, again, they collect some sort of registration fee and other things. Whether the proposed federation or consortium, having 3 or 4 or more association, members of such association will automatically qualify as members of the federation, or they have to register themselves as a new member of the federation.
To my understanding, the member of federation are the society. Society is the member of federation, and by virtue of that, each member of the society becomes the member of the federation. So this is my understanding, but maybe
that should be made clear. That should be made clear. That's all.
Yes, but for the logistics letter, but in principle, we are happy and welcome this decision. Mani Subramaniam wants to add anything. Please, Mani.
Yeah, I mean, I think it's a very good initiative. I think all the countries coming together are really seeing the light at the end of the tunnel with this kind of a consortium approach. And being in industry for the last nearly 25 plus years, worked in the US as well as India, I've actually used primarily for drug discovery and development, and it's very difficult to get a regulatory approvals for something in vitro. And the push has to come to import a lot of physiology models. It's not just small in vitro tools. You need to build more physiology-based tools, and that is missing in many of these alternative to animal conferences. And we need more physiology-based mechanisms approach. I'm working with Humane Society International in India that they're sponsoring, and I'm writing a couple of articles for them. But again, when you look at the in vitro data, I've worked since 1980s on in vitro data. But again, the complexity of biology is so high that every time I go and I say, oh, okay, this doesn't predict anything in the human, so I still have to use an animal. So we don't want to go there. We really need something that is actually predictable tools, and the regulators will automatically fall in place because you have a tool that can predict. So we don't have to work saying that, okay, regulatory doesn't ask for this, regulatory asks for this, let's do this. So regulatory will actually fall in place if we put more funding into R&D budgets to look at the human alternatives, not just animal alternatives. Okay? Right.
For the suggestion, last comment from Deepti in 1 minute if she has to add anything. Deepti Kapoor, Dr. Deepti Kapoor, please.
Thank you, Dr. Raisuddin, and thank you, Dr. Hajime Kuchma, for a wonderful presentation and all the members for very interesting discussion. My question was also related to what Dr. Michael has asked. I was interested, Dr. Hajime, that you mentioned about Asian Consortium in your presentation. And my interest was about the membership. If a society would be a member or individual members can apply, and if you can throw some light about it. That's my question. Thank you.
Dr. Hajime, are you getting this question? We are a little bit, I think you need to share the logistics and fine prints of the society confederation with the members. If you would like to add last comment, then we move to the next speaker, please.
Very good. Thank you very much. Good. So a lot of questions and or suggestion. And so we have to based on the comment, we have to continue discussion this as well for Asian communication. Thank you very much.
So now I invite the next speaker, who is a very renowned toxicologist, also involved in regulatory aspects, Dr. Chandreshwar Nath. Dr. Chandreshwar ji, are you around, sir? Hello, sir. Hello.
Yeah, we can see him.
Okay, so let me introduce briefly Dr. Nath. He's ex-scientist at very prestigious institute, CSR Institute of India, Central Drug Research Institute. He has been chairman to various committees related to the ethics. Also, he is members of institutional ethics committee of large number of institutions, which demonstrate that how closely he is working in the field of regulatory aspects of drugs development. He has been member of WHO expert committee on drug dependence. He has been regular member of several committees of Drug Controller General of India, which is equivalent to FDA in our country. So that way, we are very happy that Dr. Nath has accepted our invitation. And he is going to speak on the scope of alternative models in regulatory toxicological studies for an investigational new drug. So let us hear from Dr. Nath. And I think I mute my speaker mic. Okay.
Okay, thank you everyone. As this talk has come on.
Dr. Nath, you have to keep your mic closer to your mouth so that our voice becomes slightly louder.
Something on your publication or what I just said. Again, I am loading just a minute.
Can you raise your थोड़ा सा वॉल्यूम बढ़ा के थोड़ा पास माइक के आ सकते हैं डॉक्टर साहब नाथ जी प्लीज.
Actually, I am sharing the microphone on my desktop.
Okay. Sure. Now we can hear you. Please go on.
Yes, I am getting this sharing of screen. Yes. Just wait to that.
दो इंस्ट्रूमेंट तो नहीं यूज कर रहे हैं.
नहीं ऐसी कर रहे हैं कि शेयरिंग जो शेयरिंग है
Dr. Akbar Shah, Professor Nath requested that if his presentation can be made by your group people in sync with his talk. So can you just ensure, Professor Akbar Shah?
I think my unit is not prepared to do that. We are looking for the speakers to make their presentation.
But he sent one PPT to me also, to your group.
He did send it, but the admin is not in a position to really comply with these requests. He has been told about it. Okay. And if you could...
I think some syncing problem is there. He cannot sync with the presentation thing. And I think he has to select some window. Something is there. I think if he can work out, his team or his sons or anybody around can do that, or some children. So otherwise, If that is not the case, if in few minutes we get it, otherwise Dr. Nas can I speak extempore? Extempore बोल सकते हैं Dr. Can I speak without PPT?
Now you can see.
PPT नहीं आ रहा डॉक्टर साहब. We cannot see your PPT.
Hello, Amir. Now it has come. Hello. Hello.
नहीं आ रहा डॉक्टर साहब.
ये हमारी स्क्रीन पे तो आया है.
I think you have to share it. Some sharing problem is there.
Dr. Raisuddin sir, if you have Dr. Nath's presentation, Dr. Nath, I was trying to call you. If you have your presentation, can you forward it to me? I can upload. Amir here, sir. So this is Amir.
I am just forwarding to you. Okay.
Perfect, sir. I will upload and then I will.
Whom should I forward?
Amir sir.
अच्छा Amir. Okay. Amir जी मैं आपको करता हूँ. I am forwarding to you. Okay.
Please.
Just your this which mail I should use? CDRI wala? Amir, I sent you at CDRI email. Okay.
It is coming.
Amir, okay. No.
This I am checking, sir. Though CDRI वाला थोड़ा सा slow आता है.
Dr. Amir, I sent you on CDRI email.
Yes, I have received it, sir. I'm downloading it.
Please facilitate that. Okay.
Perfect.
Yes, some you are presenting, but it is not coming on the screen.
And then share with everybody.
Yes, I am trying.
Just. It happens sometime. Technical things are there.
In the evening, I did with the help of this instruments there, and it was okay at that time.
Okay, good, great. We can see it now. नासाब आपको दिख रहा जी? Oh, फिर चला गया. Just okay.
Okay, I will start from here. Okay.
Just we can see it here.
In the moral investigation new track. So my topic will be, I put on, now it is clear? Hello? Are you seeing?
It is visible to me. दिख रहा है. We can see it. Okay.
So actually the regulatory animal toxicity studies is required to know the adverse health outcomes in animals subjected to high dose of an IND, and with the subsequent extrapolation to the human therapeutic doses. But one of the most important, the purpose of the animal toxicity studies to obtain no observed abnormal effect level dose, because that is the key factor for the calculation of first human dose. For any new drug, it is the most important step is to calculate the first human dose, what dose could be given for the first time in phase 1 trial. So obtain a most important purpose of the regulatory animal toxicity studies. Here in the talk, I will discuss this scope and relevancy and how much 3Rs can be implemented. So this is the animal toxicology, or what we call non-clinical toxicity studies. As per the new drug clinical trial route 2019, last year introduced and the second introduced. As you know, all is a systemic toxicity studies. Then dose ranging findings, and that is dose ranging studies, repeat dose, and these are the parameters. This is nothing new. Everyone knows that. And then special toxicity studies, reproductive, genotoxicity, carcinogenicity, immunotoxicity, and local toxicity. Here you see that we use rodent and non-rodent. So now over that, what we can do for alternative to animals in these studies. So there are few tests which are already not done in whole animals. They are done cell cultures or in vivo. For example, the Hirsch channel, which is done in this cell line, particular cell line HEC293, and the binding assay are observed. This is the in vitro test. Phenotoxicity test, AIM test, it is also in vitro in the bacteria. Chromosomal aberration test, it is also in vitro. So some which are approved by the regulator in the regulatory toxicity studies. Then the alternatives. These three R's are well known by everyone, introduced by Russell and Rush in 1959. Replacement. Those are non-animal methods over animal methods wherever it is possible to achieve the same scientific aim. This is a very important point, to achieve the same scientific aim. Reductions, reduce the number of animals without compromising with the information, or to obtain more information from the same number of animals so that we should not have separate groups further to obtain them. Refinement refers to the method that alleviate or minimize potential pain, suffering, or distress and enhance animal welfare of animal users. This one point I will, when most of the discussion goes on, they are for the replacement and reduction. Refinement is also a part of the three R's. If we are using the animals and we are using a very human approach, this is also a part of animal care and this alternative part. So the alternative in vivo animal testing, we are to in vitro cell culture and in silico. Cell and tissue culture, this is in vitro testing. Samples of human cells and tissue can be used in laboratory to test cells in certain types of cells. Is beneficial because it provides cell lines originate from the human tissue, so we can extrapolate the result to the human organs.
नहीं आता हरे ने
जाता हरे
उडिंग upset mang एका की waiting एक लूसा बाला एक
on the basis of data we make some or we create a order
Admin, please mute all mics. Admin, can you please mute all mics?
Accept mine. Okay. So, based on the database, on the information we have collected, and moreover on the basis of simulation, we can use the in silico models, what we call computer models. Then some of the examples I would like to share here: skin corrosion and skin irritation test. And they have developed one human skin equivalent test can be used to replace animal-based corrosive additive studies. These studies in animals are very torturing and very painful. But now we have this epiderm formatic and epi-skin and skin-ethic RC model, which are derived from the human skin cell, which have been cultured to produce a model of human skin. These methods are currently accepted replacement in Canada and European Union in order to obtain OECD producer test guidelines which describe this procedure for the in vitro hazard anti-frictional irritant chemical. So for the skin local toxic effect, we can have a good alternative model to animals. Similarly, we have a human epidermal keratocyte model which has been cultured to mimic the human epidermis and used to measure skin irritation dermal corrosion. And very important that this test has a place, a very tortuous test earlier used by this raised rabbit skin irritation test, where the animal was observed 14 days for the erythema and edema and redness swelling or inflammatory signs. So we have got two good models which have been also accepted by the regulators for the local skin irritation and skin corrosion test for local toxicity for the skin. Another test for this phototoxicity, which is the swelling or inflammation like severe sunburn caused by exposure to light following exposure to a chemical. Here we have an in vitro model 3P3 neutral red uptake phototoxicity test. approved by the OECD detects the viability of T3 cells after exposure to a chemical presence of absolute sunlight. Actually, what we do here, there is a 3 neutral red uptake, which is a dye which is still lysosomal red, and which is more uptaken in a live cells. But if it is to exposure in a control or in the chemical treated or drug treated group to the UV light, and it is showing less uptake, that means that there is a more cytotoxicity and the candidate substance has potential phototoxicity probability. So that way we have a good alternative model for the prediction of phototoxicity, and which has also been accepted by the regulatory authorities. Then there is a very one new approach. There is human toxome. Like other, we have the signaling pathways for the so many physiological responses. So there should be a signaling pathways for the toxicity also. So on that basis, it is planned that we should study those pathways, and major component of plan include use of predictive high throughput cell-based assays of human origin to evaluate perturbation in key toxicity pathways and to conduct target testing against those pathways. The pathways approach is also used in the identification of new drugs. So that is a very good new approach to identify signaling pathways responsible for the toxicity in organ toxicities. And though of course it will be a big job, it is not a simple thing. It is more like a human genome project to understand the whole signaling pathway involved in the toxicity. But if we identify some at least, and we target, we see that if the candidate drug or IND or chemical is affecting the signaling pathway, we can predict which type of toxicity this thing can produce without using the animal. So that is a very good and new approach towards an alternative to animal models. Of course, the alternative animal models have limitation drug development, as in introductory remark. Professor Vahi Gupta has highlighted. There should be a balanced approach because there is some there's limitation and bottlenecks with the alternative model. Most important thing, we talk about drug, and drug is given to the patients in the whole body. So the test should represent the whole body responses, which lacks actually in the in vivo animal testing, and it lacks in the in vitro animal testing or alternative. Then moreover, this in vitro non-animal model cannot substitute animal model physiological response and disease, because we know in the physiological response there are so many reflexes which is present only in an intact animal or intact living system, which are not present in the in silico models or any culture plate or this. So this thing is also very important. However, non-animal cell model can limit the number of animals used in testing but cannot replace. So alternative tests cannot replace animal completely, but are unlikely to ever provide enough about the complex interaction of living system. So the living system interaction depends upon the intact system. So that is the one limit. But one of the examples is the calculation of dose. As I said, in the regular toxicity, we calculate a NOAEL, and that dose means that it is the highest tested dose of concentration of substance at which no such adverse effect is found in exposed test, or where higher dose of considered result in adverse effect. So 1/10 of it NOAEL is extrapolated for the to the human dose, and less than 1/10 fold safety margin is given to the extrapolated human NOAEL dose, and that is recommended as the starting dose or the first in human dose. So this type of dose calculation is not possible from the alternative in vitro silico or in models. So just to conclude or summarize that scope of alternative testing in drug development, how can we place alternative model where and where we can't place or have limitations. There is a discovery phase for every drug which is in the lab. Here is a big scope of alternative models. From that, we can identify the active leads by in vitro test, by in silico modeling, whether it is a chemical model, designing of the molecule or testing on the receptors, target or protein. So here it can play a big role in identifying the active breed. But once the active breed is identified, it goes to animals. For reason pharmacology, we need effective dose. Without effective dose, we cannot proceed further. In PK, we want to know half-life and tissue distribution. That can be in the whole body system. Then I earlier told you, toxicity, that is the Noel doses. So calculation of dose, tissue distribution, and this we need animals. But however, we should encourage the use of non-animal model wherever possible. And when animal model is must, the fewest possible number be used with utmost human care. This is also part of the 3 R's must. And from there, then we go to translational phase to the patient, and if it works and patient gets relief, we call successful drug. Now there is a big question, and so many times debate is there. Why we go to this part? Why cannot come to from here to that we are patient? For that answer, no, we cannot approach this. We should place alternative in the discovery phase. We can make a full use of that, and that will reduce the time. That will give us very good predictions and animal systems. And here we can use wherever it is possible and with the least number of animals. But as I said, there is a balanced attitude in the drug development, because we have to keep always in the view we cannot take this for the. So with that, we have a balanced attitude and alternative revival over body model.
Thank you, Professor Nath, for this very important lecture. There were some technical glitches, but anyhow we made it. And now, if Professor Akbar Shah would allow, or Professor Y.K. Gupta would allow 1 or 2 questions, if there are any pertinent. Otherwise, I hand over for this next session. Professor Gupta, can we allow anybody?
Maybe we can allow
one question if any participant can ask, because this is a very important issue. Ultimately, all things have to go to regulatory level, which is meant for drug development and also have industrial issues. So please, if one question, otherwise I thank everybody who has joined. Any one question, please? Please. Hello?
Technical glitches, and YK has said that alternative method we have developed in Corona, that this video conferencing. So the same alternative.
Thank you for this nice talk. Now I hand over this session to Dr. Akbar Shah to make the next announcement, please.
The chairperson for the next session is Dr. Adip Roy, one of our joint secretaries. Is Dr. Adip Roy around?
Yeah, can you hear me?
Yeah, we can hear you. Yes, you are.
Okay,
we see you.
Great.
Yes.
Okay.
So the session is now handed over to you.
Okay, great. Thanks a lot, Dr. Akbar Shah, and thanks everybody. And well, this session has got eminent speakers in it, and I think all of you would be aware of them as well. And Dr. Subramaniam Vangala and Dr. Deepti Kapoor has been a part of these alternate meetings for the last couple of years as well. And Dr. Mohanan has been a stalwart in the area of toxicology. So just to introduce Dr. Vangala, he is the Chief Executive Officer of Reagene Biosciences, and an experienced pharma scientist with more than 2 decades of experience, has got many awards and has worked in a lot of international companies as well as in Indian CROs, and has got more than 50 publications to his credit. So without wasting much time, I would like to request Dr. Vangala to go ahead with his presentation. And it's in vitro surrogate models for vaccine development and drug preparing for SARS-CoV-2 induced by COVID-19 disease. Over to you, Dr. Vangala. Dr. Vangala, I think you're on mute. So if you can just unmute yourself.
Okay, unmuted. Can you guys hear me?
Yeah, we can hear you, Dr. Wangala. Great.
Yeah,
We can
okay.
hear, we can see.
I'm going to how do I do that here? Now, right? Okay.
I mean, if you just go to the bottom of the screen, you'll see present now. I mean, you
Yeah,
can.
I got it.
Yeah, and you can just share your desktop. I think that would come up.
I'm trying to share, but the share is not highlighting. Me?
It's not coming?
The share button is to share it.
Do you see present now button, Dr. Mani?
Yeah, there's a blue share button, but it's not really.
Not the share button.
Now.
Just present now.
Yeah, I did that, but do you see any screen right now?
Sir, kindly click that image and then click share, sir.
No, first.
Sir, kindly click that image, then the share icon will be enabled, sir.
You have to click your presentation and then it will come automatically.
Sir, after clicking the present now button, click your entire screen and then you have to click that image displayed. Then the share button will be enabled, sir. So can you see the wizard of share your entire screen? So can you hear me?
No.
Sir, very first you have to click the present now tab. Sir.
I did that.
Sir, then your entire screen. Click on your entire screen.
Okay, got it. Yeah.
Then click on that image. Yes, sir.
Yeah, it's coming.
Yes, sir. Thank you, sir.
That's it. Good.
Hear me clearly now, right?
Yes, we can. Dr. Wangala, please.
Yeah, very. I think had doing except for COVID-19 business. Even that in India also that most businesses actually survived when they switched to COVID-19 related products and research. Takes 5 to 7 years to do any vaccine development. Thanks to this year, I think Pfizer, Moderna, and all announced the vaccine.
I think there is a sound issue.
Can you hear me now?
Can hear you, maybe.
It's
internet
breaking in
speed may
between.
be also slow. I think because of the internet speed, sometime.
Yeah.
Shall I turn off my video?
Dr. Mani,
Yeah, you can turn off your video. It might give you a little bit more bandwidth and.
No, he cannot turn off video because he is presenting.
Yeah, I guess.
So I think we have to bear with
Okay,
that.
I turned off my video. Okay. Yeah. Now, can you see the screen again?
Yes, please. We can do.
So
We can hear you also. We can hear you and see also.
So you guys know that viruses are pathogens. I mean, like even bacteria, even many drugs also actually perturb the immune system. Environmental pollutants as well do that. So what does happen in the immune system, overall immune system? That immune system has several antigens. Okay, a virus like SARS-CoV-2 has probably, as I understand, more than 30 antigens. So you're imagining 30 antigens, maybe 100 antibodies for each antigen. Anyway, there are two different of immunities, innate immunity and adaptive immunity. Innate immunity is actually almost immediately response, and adaptive immunity takes a while to respond because the antibodies need to be produced. And these are the two, but they do talk to each other because innate immunity comes to rescue first and stops some kind of a cytokine storm or some other pathological event that could aggravate the situation. In COVID diseases, the first barrier is that skin or nose, lungs, pharynx, gastrointestinal tract, genitourinary tract. These are all first defense mechanisms. They are barriers to not allow the viruses or any other pathogen inside the body. And you see that there are other, in fact, within that, the mucosa, acid, epithelial cells, normal healthy flora, they all contribute to that protection of the pathogen. Now, the other one is the complement activation. Complement activation is called humoral pathway immunity. And this actually is a very interesting pathway that sometimes, if this is perturbed, there's something called C3. C3, there's some issues sometimes. It can also undergo to anaphylactic shock. So always these immunities don't help you too much. Sometimes they do, sometimes they don't. So there's a membrane attack complex that forms and that activates the complement. And that's a series of pathways that is, I'm not going to go into the details. And the immediate response with cellular immunity, the neutrophils, dendritic cells, they also, what they do is they get activated immediately, and those activation actually can phagocytose the viruses and other pathogens and kill them directly. And the viral debris gets excreted out. And the adaptive immune system is the real long-term memory-based protecting system that's primarily based in the lymphocytes in the blood. There are two types of lymphocytes, T cells and B cells. When the T cells have helper CD4 and killer CD8, T cells do activate B cells, and the B cells produce the antibodies. And the antibody production can take sometimes a week or maybe 2 weeks, depends on the immune competence of the person. The other problem is sometimes the antibodies may not be neutralizing. Yeah, they are produced, but they may not neutralize the virus or other pathogens that are involved. And if you look at the SARS-CoV-2 virus, major ones, I'm not talking about all antigens. So far, major antigens that are identified include the spike protein. That is the primary one people have been focused on because that is the one that actually binds to the ACE2 target on the lung cells and activates the internalization process. And you also see membrane proteins are there, nucleocapsid proteins are there, and RNA is there. And there's an envelope protein. These are all the major antigens people actually are currently working on to see if they can get vaccines or antibodies against them to treat COVID disease. So, but there are lots of immune compromised patients are also there. The thing is, everybody is not immune competent. Why is that? That's the beauty of nature that we live in. AIDS patients, of course, they are immune deficient. Patients undergoing chemotherapy, usually blood cells go down or produce too much. Some types of blood cells may be produced too much during chemotherapy. And cancer, of course, it is also a lot of cancers do activate the factors that actually downregulate the immune system. And autoimmune diseases like lupus, arthritis, Crohn's disease, Graves, etc. All these things also contribute to the immune compromised status. And also children born with rare disorders present at birth also have a poor immune system that can fight the pathogens. Many drugs, in fact, corticosteroids, tumor necrosis factor inhibitors, and anticonvulsants, etc., have been shown to reduce the immune system competency, and that's a well-known factor. And chronic diseases, diabetes, kidney disease, hepatitis, alcoholism can inhibit the immune system. And finally, the aging. Of course, we were born with a very good immune system maybe when we were born, but as we age, we produced fewer T cells, macrophages, and complement proteins, so we don't fight the infections very well. And that's also a contribution to poor immunity in our body. So it's very difficult to say who's immune competent, who's not immune competent in the populations, because you don't know what hidden factors contribute to the poor immune competency. So typically, active versus passive immunity in therapies people have used. Active immunity is for healthy individuals, and passive immunity is for immune-compromised patients primarily. So anybody who has an active immune system, the pathogens can be neutralized very rapidly. They don't need a vaccine. They're blessed people. And also, the vaccine can also produce antibodies without any other harmful effects, and also can create active immunity. And the passive immunity, immunizing directly with antibodies derived from the other sources. Of course, people have used rats, mouse, camel, llama, chicken, monkeys, and also now people are trying to use in vitro systems to produce antibodies, not using animals. So if you look at the pathogenesis of SARS-CoV-2, the primary binding site is ACE2 receptor, but also an accessory protein called TMPRSS2 that aids in the internalization of the SARS-CoV-2 spike protein into the lung cells. Once it's gone, it actually activates the angiotensin II system or direct viral invasion. You're actually seeing another problem is the cytokine storm. The cytokine storm is actually part of the innate immune system that is supposed to stop the viral spread and inactivate the virus to some extent until the adaptive immune system kicks in. But the problem is sometimes cytokine storm is overactivated. This is what's happening in SARS-CoV-2. The activation is too high, and in fact, it doesn't give in time for you to activate the adaptive immune system. This is where many deaths are seen because of cytokine storm is too severe in many people. So cytokine storm, which is supposed to protect, actually becoming a fatal cytokine storm. And on top of it, all these things also contribute to the hypoxia, hypotension, and everything. And then you have a series of things also happening, rhabdomyolysis, acute cardiac arrest, and also sepsis. Sepsis is another interesting thing, is like multiple organ failure. So, in fact, there were some studies done on age-dependent pathogenesis of human coronavirus. But this is not about the SARS-CoV coronavirus. And if you can look at it in healthy young individuals, you can see that the protective cytokines like interferon, alpha, beta, IL-12 are actively fighting the infection. Whereas in the aged individuals, in fact, they are lower in number, but the anti-inflammatory cytokines, and actually inflammatory cytokines, I would say, are higher, TNF, IL-6, MCP1, MCP3. In fact, many drugs people are trying to use right now are IL-6 inhibitors to try to stop the cytokine storm to some extent. So what's happening is...
Dr. Wangala, would you like to wrap it up in a couple of minutes, please?
Sure. Okay.
Okay, great. Thanks.
So the treatment strategies that have been used so far include cytokine storm inhibition, with several drugs that have been used, viral replication, vaccines for adaptive immunity, and antibodies for passive immunity, and antibody drug conjugates, combination therapy. You can conjugate an antibody with a drug, and so you can also do drug-induced therapy as well as antibody therapy. Now, I think, unfortunately, today we do not have much of luck with using in vitro tools, but they're under development. In fact, there is some progress that has been done with some antibody therapies. And we at Reagent Biosciences actually also developed the 3D lung vascular model for rapid drug repurposing as well. I'm not going into this, there's a paper published in Nature Methods and how they use rapid animal-free antibody generation to SARS-CoV-2. They have published excellent data showing that the animal-free antibodies are actually working very well. They are protective antibodies and they're neutralizing antibodies. They're also, there's a huge library that's also created. If you want, you can also look at this library and pick and choose your recombinant antibodies that you want to synthesize, either monoclonals or polyclonals. And the final one here is that antibody-free antibodies are better neutralizing than animal-free antibodies are better neutralizing that. The clinical animal sera, most of the time, they don't have neutralizing antibodies. People have done the convalescent plasma, not very successful. So animal-free antibodies can substitute for that. So drug repurposing is faster than traditional drug discovery application because most of these drugs that have been already used have been approved for other indications. Though there are not much of safety issues that you need to wonder. Maybe small minor toxicology studies maybe need to be done depending on the route of administration. You have pharmacopoeia database, maybe about 3,000 drugs in there. You can use artificial intelligence and do the QSAR analysis and identify potential molecules that might interact with the various targets I already described before. And screen all these molecules in our 3D lung vascular model. And this is limited rapid preclinical testing. And I think under the conditions, regulatory agencies would approve very, very minimal preclinical animal toxicity testing. And so you can go to rapid into clinical testing and submit your dossier to regulatory agencies. And this is what we are working right now. And we also have other programs that we are working on SARS-CoV-2 for passive immunity. I'm not going to go into that details, but this is right now, we have used alternatives to animal method, trying to come up with a drug repurposing model within the reagent. So the essential summary and conclusions include that unfortunately there are no alternative approved models currently for vaccine development. The regulatory path requires animal models. In the absence of regulatory framework, rapid alternative in vitro tools need continuous R&D funding to replace animal models. Drug repurposing is faster approach as many molecules were already approved for other indications and limited preclinical testing, if any, is needed. So combined approaches of artificial intelligence and drug repurposing approaches can speed up the drug discovery process for SARS-CoV-2 treatment, and recombinant antibody developing using in vitro models can also help substitute vaccines for immune-compromised patients via passive immunization. We are also looking at antibody drug conjugates for SARS-CoV-2. We've already tested them in lung model, and some of them are working extremely well with the antibody plus drug conjugated model. I can't go into the details because of proprietary information. So thanks very much. With the limited time I have, I could speed up the presentation and present the salient features of how you can use alternatives to animals in drug SARS-CoV-2 or any other viral pathogens, etc., that we need to contain in the near future. Thank you.
Thanks, Dr. Wangala, for this educative presentation on pathogenesis related and animal non-animal models on that. Any one quick question?
Yeah.
And then we can move on to the next speaker. Anybody has a burning question?
Dr. Wangala, can I ask you one question?
Yeah.
Hi, this is Dr. Pratiksha here. Very interesting presentation. Thank you so much. I just wanted to know, what was your rationale for choosing the cell line, or maybe the 3D model for COVID specificity 3D modeling? Means, what is the objective of how did you go about?
Yeah, this is human lung having high expression of the ACE2 receptor. Without ACE2 receptor, the SARS-CoV-2 doesn't get inside. So we're looking at ACE2 inhibition with drugs and antibody drug conjugates as well. We are also going and testing the lung model with ACE2 overexpressed models. And unfortunately, we don't have the SARS-CoV-2 virus. We cannot, because you know how virulent it is. So we are also using pseudoviruses trying to test the other models. But primarily our usage is on lung vascular model, 3D vascular lung model, which also presents some vasculature there to see if we could look at the blood clotting features and other things that have been reported in SARS-CoV-2.
Thank you so much.
Yeah.
Great. Thanks a lot, Dr. Wangala. And if anybody else has got any questions, they can post it in the chat box in the message center. I think it will be visible to everybody, and Dr. Wangala can respond to that. We'll quickly move on to our second speaker, Dr. Deepti Kapoor. She is the policy advisor with PETA India, and she's skilled in molecular biology and has a strong professional background in medical biochemistry. She has conducted her thesis in medical biochemistry, understanding of TPH genes in suicidal behavior. And she would be presenting on COVID-19 poses that we cannot waste time on animal tests. Dr. Deepti Kapoor, please. Over to you.
Dr. Adib, for the introduction. And good evening, everyone. I first would like to thank Dr. Akbar Shah and everyone for giving me this opportunity. I have been facing some issues, so I'll switch off my video and then I'll start my presentation. I hope everyone can see my screen.
Yeah, we can see.
Great. So, as Dr. Adib Roy has already introduced me, I work as a science policy advisor with PETA India. It's People for the Ethical Treatment of Animals. And many of you may already know, PETA India is a non-profit organization, and our primary area of interest is animal protection. But we also value good science, which benefits humans and animals alike. And we work to focus on advancing good science that does not include animals. And in today's presentation, I'll focus on work during the pandemic in support of our mission statement that in part reads that animals are not ours to experiment on. And as you can see from the slide, we and our affiliates around the world, we do have scientists on staff who work full-time to encourage the use of robust in vitro models to replace and reduce the use of animals in research, in teaching, and in regulatory testing. It involves everything from funding method development to holding informative workshops and webinars, meeting with regulatory agencies and companies, and participating in scientific meetings and conferences as we are here today. So quickly coming on to the topic. So we know that the use of animals to try to understand human disease or to predict human responses to substance has long been the dominant paradigm in biomedical research and regulatory testing. Yet as time passes, we are becoming increasingly aware of the failure of animal methods to establish both efficacy and toxicology risk, and the inherent problem associated with the extrapolation of data to human populations. Dr. Y.K. Gupta in his introduction has very well and very correctedly mentioned this. There are many places where it doesn't work. Now, astonishing advances in research technology are beginning to revolutionize scientific policy around the globe with the transition towards human biology-based methods. But transition takes time, and there are many national and international agencies and funding bodies that are yet to make a move. And we all know that there are many factors to play in the failure of animal models to predict human outcomes reliably, including reporting and publication bias, poor study design, and inadequate sample size. However, the weakness of animal experiments cannot be overcome by simply improving study design, because critically intrinsic biological and genetic differences among the species, they contribute significantly to inescapable translation problems that we face when we discuss about translation from animal model to human model. And also that there is growing scientific consensus that far more is to be gained to understand and develop human relevant research methods and technology than from reliance on animal studies. With this in mind, we know that regulatory agencies around the world, though many have yet to make a transition, but many regulatory agencies have started policy changes concerning replacing the use of animals in regulatory tests and biomedical research. From this time, as I've discussed, that's a paradigm shift, and we do have to keep time because this shift is going on. These are the headlines that were seen in the press within last 3 to 4 years. However, it is to be noted that headlines can be misleading. So let me provide some context to these headlines. In the year 2016, the Dutch government announced its plan to phase out toxicology tests for chemicals, food ingredients, pesticides, medicines, whether for human use or veterinary use, including vaccines, that they will phase out these toxicology tests on animals by the year of 2025. And this decision was after the Dutch National Committee for Protection of Animals Used for Scientific Purposes, NCAT, where this agency stressed that the need for paradigm shift away from treating procedures on animals as a gold standard. So we're not just talking about ending all tests on animals in the Netherlands or the Dutch, but the proactive policy taken by the Dutch government is a tremendous step in the right direction. Then the Chinese government has now approved 3 non-animal tests for testing of cosmetic products, which will spare countless animals the agony of having their substances forced down their throat or applied onto their eyes or on their skin. In August 2019, the year 2019, the PETA US Director of Regulatory Testing, Dr. Amy Klippinger, she sat beside Dr. Andrew Wheeler, Administrator of the US Environmental Protection Agency, at the historic signing of memorandum outlining EPA's commitment to stop funding and requesting tests on mammals by the year 2035. And the short-term goals identified to meet this goal by 2035 is the commitment to include reducing tests on mammals by 30% by the year 2025, and providing researchers who are working on development or developing these non-animal methods with substantial funding. And this was a very fantastic news and a very good step indeed. Now, India is not far behind making these changes. And based on a paper published in 2019, the Indian Council of Medical Research, ICMR, and this paper authored by Dr. Soumya Swaminathan, they actually pursued and acknowledged that there are emerging technologies such as organ-on-chips, microphysiological systems, and other methods which can effectively replace use of animals in experiments. And they also appreciated and acknowledged the fact that although the move would require a costly overhaul of drug approval process, but recognizing that non-animal technologies can outperform animals in their ability to model human diseases is something that is very much appreciated and is very much documented in the Dr. Soumya Swaminathan's paper. And in addition to their documentation, they also proposed calling to government to establish centers for excellence for developing such approaches as to increase funding in funding options nationally and promoting international collaborations. Coming to the present scenario, past 2020, we all have seen the effect and impact of spread of COVID-19 cases around the world. And it was very well felt and realized that we need effective treatments and vaccines, that we can't waste time on ineffective measures, procedures, protocols, or bureaucratic pathways, which also include testing on animals. And lucky for us, the authorities responsible for approving medicines, they agree. And in a watershed moment in history, the medicine regulators around the world, including India's Central Drug Standards Control Organization, the European Medicine Agency, the United Kingdom's Medicine and Healthcare Product Regulatory Agencies, the Netherlands Medicine Evaluation Board, and the US Food and Drug Administrator, all these regulators, they have responded to the pandemic by opting for human clinical trials over animal testing. And PETA India and our affiliates, we are asking the Indian government and other regulators as well to continue the good work by prioritizing investment in human-relevant non-animal therapies to fight COVID-19 and other diseases. And despite the existence of human-relevant non-animal tests, medicine regulators have earlier insisted, for more than decades, they have been insisting that there is no way to bring a new drug to human clinical trials without first testing it on thousands of animals. But in the face of pressure to develop medicines to prevent or treat COVID-19 quickly and safely, these agencies have taken actions proving that this is not the case. In early April of 2020, for the first time in the history, both the United States FDA and US National Institute of Health, they greenlighted a landmark human clinical trial for vaccine without first requiring extensive animal tests. Companies are usually required to spend years of putting animals through the agony of new product test before human clinical trials are allowed to begin, even though those animal tests do not predict whether a drug will work in humans. But in this case, a company started developing COVID-19 vaccine in January, and by March, the US FDA and NIH both agreed that it's time to see whether this vaccine is effective in humans, thereby surpassing the testing on animal step. And this was just the tip of iceberg. Soon, many similar trials were up and running into planning stages. And we all, I believe we all have been following about the development of vaccine and the other stages and the parallel studies that are going on. Now, concluding, in addition to the present scenario, avoiding misleading tests on animals and asking human volunteers to give blood have sped up access to potentially life-saving treatments for COVID-19 in the very initial stages, and it's still going on. The most readily available treatment for it and the other diseases, which is the convalescent plasma, and a lot of details about the recombinant antibody production and antibodies has been covered by Dr. Vangala, so I won't go into much details about it. But basically, convalescent plasma, it comes directly from humans who have recovered from the illness or have recovered from the illness but still carry the antibodies. And this treatment, extracting antibodies from the plasma and then using it as a therapy, does not use any single animal, just the antibodies made by the human immune system to fight off infection. Now, the government has already allowed several people to receive convalescent plasma as part of its treatment for COVID-19 and is also expanding its effort. And there is a modern technique called Phage Display, which can be used to produce human-derived antibodies on an industrial scale to treat infected patients without any continual dependency of supply of convalescent plasma from patients. So this is a modern step that we have been requesting the government to look into. And Dr. Vanghale has already quoted one of the papers from Wenzel 2020, which is a joint effort of PETA International Science Consortium with our international collaborators to develop such therapeutic antibodies for treating diseases like diphtheria. And the same technology and the same principle can be applied for developing treatments for other diseases as well. Now, to help the government update the policies and put forward to come up with the policies and a plan to how to effectively replace use of animals, as Dr. Y.K. Gupta has also mentioned in his opening remarks, that we need effective plan to come up with replacements rather than and effective approach. So to help government and researchers to get an effective approach to how to bring in policies and changes for replacing animals with non-animal, more effective and humane methods, we have come forward with a strategic plan of action proposing strategies for prioritizing, modernizing research and testing in India. We have developed this tool and we call it as a research modernization deal. It is a very beautifully made report tool. That
Dr. Deepti would request you to, if you can.
Yeah, I know.
Sorry about that. Yeah, thanks.
I'll just take 2 minutes. Thank you. Yeah. So quickly going through the research modernization deal. If you are interested in how what this deal is, and if you want to get a copy, you can email it to me, or you can check our website. So we have identified 5 strategies to end use of animals in experiments. One is to immediately eliminate animal use in areas which animals have already been shown to be a poor and unreliable predictors for human models and have been impeded. So we have identified the sections in the report. Second is to conduct critical scientific reviews to identify the areas in which the use of animals can be ended immediately. The third step which we have identified is to implement an ethical harm-benefit analysis system through retrospective evaluation, and not just to have a checklist or a check mark, but to have transparent evaluation. So we all know the Committee for the Purpose of Control and Supervision of Experiments on Animals, that is CPCSCA. It has certain procedures where it evaluates and permits the use of animals. So having a better evaluation plan of those protocols to actually see whether the previous permissions have been successful in providing or completing its purpose, or does it need another approach. So those strategic plans are there. Then global harmonization, that is acceptance of data and methods which have been developed abroad through within the countries, nations, and organizations. So OECD is one process. Getting OECD approved methods adopted in India is one process. We have been successful in getting OECD approved method considered for Dre's test. Dr. Y.K. Gupta was the head of the committee in Dre's test, and I was also one of the member of that committee. Then we have also been successful in having several harmonization with respect to pharmacopoeial test. So these things, other development is required. I'll just take one more 30 seconds more to complete this. And then is the funding aspect of developing new methods and identifying the places where development is required. The Indian government has announced that in the very beginning COVID-19 Research Consortium and other initiative for funding research on ways to prevent, treat, and control the disease, and is actively investing. So a proper approach and diversion of funds from animal model-based studies to non-animal model-based studies, including but not limited to in vitro model, in silico models, big data analysis, microphysiological systems, and all that is required, and a proper guidance is required. And with greater investment in exciting and innovative non-animal methods and bold policy initiatives, far more promising cures and treatments for humans can be developed. And we have witnessed regulatory agencies and companies abbreviating testing on animals and preferring first in human clinical trials to get treatment for COVID-19. And what we are now working and promoting and suggesting everyone is to continue this effort. And with that remark, I conclude my presentation. I'm very sorry, Dr. Roy, for exceeding the time limit, but let me know if you have any questions.
No worries. Thanks a lot, Dr. Deepti, for this presentation. And I'm talking about the policies that are, prevalent globally and being taken forward for, animal removal of animal testing. So in the interest of time, we can directly, Dr. Akbar Shah, we can directly move over to the next presentation. Is that fine?
I think so, because we can take later. Deepti,
we can take the questions
talk
later.
about very exciting, so we can
go to next speaker.
So our next speaker. Our next speaker is Dr. Mohanan. So I think all of us know Dr. Mohanan pretty well. He's a fellow of National Academy of Sciences, also fellow of the Royal Society of Biological Sciences, UK. And his list of accomplishments is quite long. I won't go through it except for the fact that, he has been sitting on many government committees as an expert, especially in the development of document for vaccine development and licensure for COVID-19 and review committee on genetic manipulations. Dr. Mohanan, over to you. I've kept your introduction pretty brief, so you can take up that time in your presentation. Thanks.
Thank you. Hello? Is it audible?
We can hear you. We can hear you.
Some sharing is problem. Yeah.
I think your presentation is uploading.
I can't see here.
So you can go to present now if you are able to. Yeah, just 1 minute. You can click on the presentation again and it might just
1 minute, just 1 minute. But it is coming something. Stop presentation again.
Yeah, present now.
Present now. Then go to the entire screen.
Or you can see your desktop. Does it say desktop? You can click your desktop.
And desktop I can see, but my presentation I can't see that. That's the problem. Oh my God.
Now, once you click on the desktop, then you can choose your presentation.
Yeah, okay, then it is there in the desktop. So my presentation is there in the desktop.
Sir, kindly click your entire screen on present now.
Yeah, I did it.
Sir, then
Just
click on that. Sir, click on that image on your share your screen.
I did it.
Then the share button will be enabled, sir.
Yeah, then I did it, but it's your presentation.
Then minimize this wizard and go to that desktop, sir. Sir, don't press stop sharing, sir.
No.
Can you minimize the wizard?
I'm again going. Okay, so in your screen, then I can see my thing. So share it.
Yes, sir.
Yeah, but it's not coming.
Mohan, have you kept your presentation open?
Yes, it is open, sir.
Then
Sir, minimize this page and click on that desktop, sir. You can click on the PPT, sir.
Is it able to see?
Yeah. But
Okay.
it's not full screen.
Is it okay?
Yeah, now it is full screen. Yes.
Okay. Good evening, all. I'm sorry that there was some communication gap. It's a technical failure from my side. Okay. So I'm going to present an update on the in vitro pyrogen assay, which we developed indigenously from our institute, and also a humanoid chip technology. This is the recent advancement in the biology, in the biomedical sciences. So I'm not going for an introduction. I think our institute, which is coming under Department of Science and Technology, Government of India. We have 3 wings. One is the biomedical technology wing, where we are developing all technologies, and this is the tertiary care hospital for cardiac and neuro specialties, and this is the Achyuta Center for Health Science Studies. And the objective of the biomedical technology wing is the promotion of biomedical engineering and technology, where the main operations are the medical device development, medical device evaluation, applied research activities, and human resource development. And you know that we have developed around 41 products in the market, and which has the technology has taken from different companies all over India, from from the Himachal Pradesh to Kerala. These are some of the products, and this is the blood bag, and the blood bag which you are getting in India, also in the abroad, and 70 this blood bag is exported to 70 countries. And our heart wall is having 130,000 patients is having this heart wall as on today. Okay. And the next is, that we have several patents. There are 108 patents and patent grants. And what is our patent to commercialization ratio is 30%. So whatever the patent is there, we immediately go for the commercialization, means the technology transfer and the product. We have a technology proving facility here, and it is transferred to the industry. Okay, and these are the area we have. We have material qualification and product development, large animal studies, and product release, and it's all coming under the single umbrella. And I just want to say that this is a Pyrogen Test which we developed indigenously, which is in par with our Thomas Hartling also. He is the first person who developed such an assay, that is in vitro pyrogen assay. And so it is the basic, the program is based on that. And the objective is to develop an ELISA pyrogen testing kit from human whole blood. And this in vitro human whole blood assay will be suitable for evaluating the biomaterials, medical devices, pharmaceuticals, and blood-derived products, and a wide spectrum of application to measure the undetected non-endotoxin pyrogens, such as pyrogens of any chemical. So irrespective of chemical nature, you can detect the endotoxins. You know that we have, as on today, the rabbit pyrogen tests are the, then lal tests are there. The damage pyrogen test will detect both the endotoxin and the other pyrogens can detect. But the only in vitro pyrogen test which is available is the lal test, which will detect only bacterial endotoxin, forgetting all other types of the pyrogens. And what we developed here, we immunize the interleukin 1 beta to the rabbit and collect the blood. And one rabbit is good enough for 5 to 6 years. And collect the blood, and serum will be and the plasma will be separated. And this plasma will be purified and coated on an ELISA plates. And these are the quantity. The coating is done with the 10 microgram per ml of the primary antibody at room temperature, and then washed with deionized water, which is blocked by the 1% BSA and at room temperature. And then the next step is the antigen steps. The antigen that is at room temperature, then washed 3 times, and then antibody step is labeled antibody again, and the substrate with the 50 microliter of TMP substrate. I'm not going to explain all these things because my time is very limited. And you can see here, sorry. Yeah, you can see here, this is the in vitro pyrogen test. We have a kit. We developed a kit, and where all the reagents will be there, and with an ELISA plate. And then during your if you want to do the pyrogenicity of any devices or any chemicals or whatever it is, you can take the 150 microliter of fresh blood and 250 microliter of blood media and 100 microliter of pyrogen free, that is saline. Then make up to 1,200 microliter with that. And then you can incubate at 37 degrees centigrade for 1 hour, then 2 hours, 3 hours, then up to 8 hours. Then after that, you have to centrifuge it at 500 gram for 2 minutes, and then serum will be diluted with the 10 to 25 fold, and then you can take the reading. And you can see that this is the interleukin 1 beta range. You can see here, this is the control, is the normal thing, and this is the endotoxin, 1 endotoxin limit. There is a limit at you can get at 4.5 hours, and you can see here it's around 4th hour. You can see the maximum interleukin release you can observe. So this is in par with our the Hartling Wendell in vitro toxicology, which was published in 1996. Okay, so this is the interleukin 1 beta. This is maximum. They are also getting in the around 5 to 8 hours. And the response to endotoxin stimuli is concentration dependent, and the high concentration of endotoxin, and you will get at the earlier period. And the interleukin 1 beta release reaches a maximum within 3 to 6 hours. There is post injection, and then the negligible we can see in the other cases also. And this is the proposed kit. You can see here, this is what is that ELISA plate, which is coated plates, and it's all are the reagents, which contains the experimental manual, the 96 well plate with sealer, the pyrogen free media and saline, then endotoxin and non-endotoxin, then 10x blocking buffer, then 10x diluent buffer, then 10x wash buffer, and HRP labeled antibody, then anti-interleukin 1 beta antibody, then TMB substrate solution, and the stopping solution. And this is patented as per the Indian patent number is 314849, and this is in last year. Okay, that's about, just one more slide. In this interleukin 1 beta kit, this ELISA system and can detect 15 picogram per ml, that is up to 50,000 picogram per ml level, and detect pyrogenic potential response to 0.1 endotoxin level as recommended by WHO or FDA. The chemical pyrogen stipulated by ISO 10993 part were used to induce pyrogenicity and detect at a minimum concentration of 1 nanogram level per ml. And the interleukin 1 beta level reaches a maximum period around 4 to 6 hours. Okay, that's about the interleukin, in vitro pyrogenesis, and now I am going to the human on a chip program. And this is also indigenously developed technology, which is we are in the very successful stage now. And you can see here the objective is the development of human on a chip, and which will be mainly used for the biological evaluation of the pharmaceuticals or the devices or whatever it may be, chemicals. And the third is disease modeling using human on a chip. This is also a very good progress in our end. And these are the immediate objectives: the fabrication of microfluidic chips. This is a very important step as far as the human on a chip is concerned. And we successfully formulated 3 designs, and 1 or 2 design we are approached the patent office for design registration also. And the development of human on a chip and development of microculture on a developed chip. That's also, you know, that the culture is very important because the number of cells are very less, and the pathway or the passage is very less. I mean, it's very narrow, and which is less than the size of our hair. And it is through the microfluidic system. The passing, the peristaltic movement through the is by the by the microfluidic system. Then assessment of repeated dose toxicity using human on a chip. Then assessment of pharmacokinetic or toxicokinetic using human on a disease modeling and ADME profiling. You can see here the success of organ on a chip microphysiological tissue culture system has stimulated the researchers to challenge a more systemic level in vitro human biology through human on a chip system. This system aimed to combine several organ equivalents within a human-like metabolizing environment, or aim at in vivo-like pharmacokinetics. Also aim to extend the capability of a platform towards the generation of repeated dose exposure and ADME, that is absorption, distribution, metabolism, and excretion profile. The ADME profile is crucial to analyze if a drug reaches its intended target and has a therapeutic effect or not. You know that the drug development will take a minimum 10 to 11 years, and the preclinical stage itself is 1 to 3.5 years. And to minimize this period, as well as to minimize the animal, this is the only technology you can use it now, and people are going for such type of devices. This is a state-of-art technology. The concept of human on a chip have evolved over the past few years from a conceptual idea to a possible alternative for animal models. And the potential of the system is now universally recognized by the scientists, the pharmaceutical industry, and government authorities. What we are looking is, we will make a chip over that different organs, the miniature organs we wish to develop, and then we will expose the chemicals or whatever the intended area. So I'm just passing to our experience here. So the first thing is the design and modeling of human on a chip. You can see this is the first model, which is size of 2 centimeter in the width, and 4 centimeter is the size of this device. And total 7. You know that our histopathology slide is around 7 centimeter, and this is the first model we can. And here we never mind about the organ size, but in our latest the design, which we are concentrating the size of the device, as well as the flow, as well as the diameter of the passage also. And you can see here, this is the thing. Here you can see this is the width is 2.5 mm, and here you can, this is the 5 mm. And here you can see, here is the well where we have to culture the cell, and at a stretch you have to, this is for the 10 organ there. So this is the first one, then second, third, fourth, fifth, sixth, 7, 8, 9, 10. So this is a 10 organ system, and this is a model, a design which we made in a software. And here you can see, this is the PDMS picture. You can see this is a well, and over that we used to put it. And this is the passage you can see here. Okay. And I'm going to the next slide. You can see here, this is the organ, then 10 organ, and this is the flow path. And you know that this is the flow rate from here. And once it completes the passage, the fluid system will take an equivalent to our blood flow speed. And this will take a complete rotation around 4 hours. You can see here, this is 78, 85, it reaches here. And once it completes here, is a one rotation, it takes around 150 minutes. Yeah, 150 or 160, yeah. Around 180 seconds, that means around 6 hours will take for one rotation, which is equal, this speed is equivalent to our blood flow. So this is the device, this is a simulation of human on a chip. And you can see here, this is our developed system here, and we have a microfluidic pump here, and which is attached to an elbow software. It's a computer, and this is the media reservoir here. We have an inlet and outlet, and this is something like a rotating, right? And in our body system, once it enters in our blood, this will circulate all over the body. So similarly, this is the 10 organ system. So this is the representative organ. So you can organ the heart, lung, liver, kidney, and all organ you can develop. And this solution will rotate through the microfluidic pump. Okay, and this is the model here. Okay,
Dr. Mohanan will request your, kind of.
It's over. It's over.
Yeah,
it's over. Just 1 minute. Yeah. So this is the model. Here you can see the blood is flowing here. The experiment is going on. It takes around 4 hours, and this is the system. You can see here again. So this is a cell culture. You can see here, this is 10x. It's an alveolar cells. That is A549 cells we grow it. And this is after 24 hours, the cell is growing like anything. So this is 20x things. And the advantage of this technology is, one is the 3L principle. The other thing is, we all use the animal models. What is animal model? Animal model is having a systemic, but not human beings, right? It's a systemic response with the animal cells. And we have a static 2D and 3D human cell culture, but it is human, but not systemic. But in the humanoid chip, the important thing here is the human cells with a systemic response. That's the beauty of the system, what I want to tell here. And the chip is format of standard microscopy slide. The tissue culture is around 100,000 fold smaller than the original organs. Rapid prototyping of any reliable chip design, then it's compactable. It's all like this, very cost effective and replacement of this. And I want to show you these slides. I'm sorry, you can see here 2025 is the cutoff date for the US FDA for developing this. So validation is going on from world over, and including the US FDA, and in India too. We are ready to, this is 2020, we developed 3 designs which are very good in functioning, and we applied for the design registration, and 25 within 10 years, and this will be what I can say, validation will be over, and in par with the US FDA. So thank you so much.
Thanks a lot, Dr. Mohanan, for this informative presentation. And we can have a couple of quick questions for Dr. Mohanan or to any of the speakers in the session. Please go ahead.
I'm Surat. I just had one question. Am I audible?
Yes.
Yes, please.
Very nice presentation, Dr. Mohanan. So I just had one question. So in your schematic of how the chip was designed, so for example, 2 organs, 2 systems were connected together, and the flow was happening in 2 systems simultaneously, which was going to the next system, and two organs were connected. So is that a design strategy, or is it how it happens in vivo, or so why was that chosen?
No, this is a confidential thing. The thing is, I can just explain to you, all the 10 organs we connected. The thing is, there are 3 strategies. One is the cells on gels. The other is on the micropores. And you can have one layer of endothelial cells over that you are in the cells of interest. And the passage, the flow will be with a single cell. The important thing here is all the media will not function, function to grow all these cells. So we have to, our challenge is to identify a single media to which is suitable for all these cells. So we are in success of that, and also our design is in such a way to what is that, to promote the growth.
Sure.
Right. This is Dr. Mani from Reagent Biosciences. A wonderful presentation. I just have a couple of technical questions. The problem here is that you're putting organs. How are they? There's no vasculature in these organs, right?
It's a microfluidic pathway, sir.
Yeah,
peristaltic movement, and it's the passage, the flow of the fluid is equivalent to our blood flow in the body.
No, that is, I agree. But the thing is, typically any organ. Okay, right.
So there is a breakage, your sound.
All organs have blood vessels that circulate the blood into the interior parts of the tissues. So how do you ensure?
So the thing is, so I can very what is that? The first layer of endothelial cells.
I think Dr. Mohan, this is a discussion which can take over later. Okay. However, I would say the organ on chips have several caveats, and that's why it is not the final answer, but this will definitely reduce much reduction in the animal experimentation and will give an early indication. I will not say early confirmation. This will give an early indication of the efficacy and the safety of any product. So these organ on chips as on date are currently considered as a very good model for early indication and reduction in the number. They have caveats, and these caveats are gradually one by one are being handled over. So I think we can take this discussion later.
I just want to conclude saying that from 2003 we've been trying this, but the problem is you don't really get that physiological blood flow into these organs, and that is very critical. Zonal compartmentalization in liver, you can never reproduce just by putting tissues in these compartments. That's what I'm saying. Okay.
Perfect. Great. Thanks a lot for all the comments. Over to you, Dr. Akbar.
I much appreciate that.
Yeah.
Over to the chair.
So I would like to thank Dr. Raisuddin, Dr. Adip Roy for chairing the sessions and the speakers, Dr. Nath, Dr. Kojima, Dr. Mohanan, Dr. Subramanian Wangala, Dr. Deepti for their wonderful presentations and addresses. Especially, I was excited hearing the lecture of Dr. Mohanan, a kind of lecture which I've been longing to listen ever since I started Mahatma Gandhi Center in 2009. I'm glad that India is not lagging behind in the in vitro scenario, 3D culture scenario, particularly organ chip and microfluidics and kind of things. Thank you, speakers. Thank you, Chairperson, for the wonderful job. And now, as agreed, this will be a break of 15 minutes. And so from now on, at 8:30 p.m., we should all convene.
We can log off and log on again?
We can do for 10 minutes if you want, so that
if you are agreeable, we can even skip the break. If
no, 10 minutes is, I think, required. We may have to attend.
Agreed, Professor Raisuddin. So we meet at 8:25, right? And don't log off. Leave it open. You can just go and come back. Okay?
We meet at 20:35.
20:25. Yes. Another 10 minutes from now.
In 10 minutes, I will cut my vegetables.
So India time 8:25 or 20:25, right?
20:25 o'clock we meet. Okay, thank you.
Yes. Thank you.
Professor Dr. P.V. Mohanan, sir. Sir, are you there?
Looks like he left. I don't see him in the list here.
Actually, his screen is visible on the thing, sir.
Maybe he stepped out somewhere. He's still okay. He hasn't turned off his screen, is it? Can the admin remove it?
No, sir. He has to stop share. Otherwise, we have to remove his name.
Yeah, you remove it. It's gone, right? So. Anybody has his phone number? Call him.
Okay, sir. We will try.
Yeah.
No, sir.
Whose number we are looking for?
Mohanan. The one just presented.
Oh.
Yes, sir. I have his mobile number. I will call and I will tell you.
Thank you, sir. Yes, sir.
But this can be done from the administrator, isn't it? Also.
We have to remove his name from the meeting, so then only we can do it. So we can't do that right now.
No, you can remove his name and let him re-enter. I think otherwise we'll waste time.
Sir, is it okay, sir? Okay, sir. We will do it. Is it okay now, sir?
Yes, I think. Now he will re-enter. I think that's fine.
Okay, sir. Thank you, sir.
Professor Gupta, I think I am the next speaker. So am I allowed to share my presentation in this break?
Yes,
sir?
I think you should. You should.
Perfect. Thank you, sir.
If Dr. Mohanan is listening, I think he can re-enter.
No, he's not there.
He's not there.
Okay.
Is my presentation up there, sir? Is my presentation visible? Yeah.
Not fully. You got to expand it. Make it full screen. Yeah.
Yes, now it is fantastic.
Perfect.
Great.
Thank you, sir.
Yeah, it's fully visible, and once chairman speaks, you can minimize. Okay.
Sure.
We are not hearing from chair.
Maybe chair would be back by 8:25. Oh, chair is there. Dr. Idris is there.
Yeah, we can see you, but
You are not audible, Dr. Idris.
yeah.
Dr. Idris, if you are not unmute, otherwise then you have to.
Oh, he's saying
I can't see Dr. Idris.
To me, Dr. Idris is visible, but I think
You're not able, are you talking? Because we cannot hear you.
Yeah. I can also not, but he's visible otherwise.
He's visible and he's not silent also.
Yeah.
Perhaps he has to log off and then log in.
Sometimes it is helpful. Yeah.
Anyway, what we can do, Dr. Raisuddin, you can chair for the time being and he can start the talk. So, I think we can start the talk and Dr. Idris can join as soon as he is okay with his audio.
Maybe 1 or 2 minutes, I think we can wait. We told about 2025. It's 2022. Maybe 2 minutes if you allow. Just maybe people will join us. Dr. Idris, we cannot hear you.
You can hear me.
Yeah, now little bit. Yeah, fine.
Yeah, actually,
now,
yes.
Now we can hear you. Fine.
Yes.
At least I can hear you and see.
Yeah. I was there, but suddenly the audio is gone.
No, maybe 2 minutes we wait because it is 2022, so sometime around 2023.
Another 3 minutes will wait.
Some people are kept on their mic, so I switch off, then others, only Dr. Idris keeps his mic on, then we can hear you very clearly. There is echo sound.
Dr. Idris, I think you are logged in from 2 devices. I think that's what is interfering in the voice.
I did.
So you might want to mute one of these. Yeah. If you mute one of these devices.
Yeah, I did.
Yeah, perfect. Then it will be good.
Professor Akbar, can you announce resumption of session?
Yes, sir. So we reconvene the technical session and the conference, and we have now the third technical session to commence. The chairperson is going to be Dr. Muhammad Idris, a senior principal scientist, Center for Cellular and Molecular Biology, is the famous CSIR Institute from Hyderabad. So over to Dr. Idris. You have 3 speakers and please maintain the time, 15 minutes each.
Thank you. So welcome to the international webinar and third national conference of the Society for Alternatives to Animal Experiment. So we are half done for this lovely evening. The happy note here is it's very happy to see the alternative biologist or scientist meeting in an alternative way for an alternative normal, connecting scientists from several countries in a single point of time. So before starting, I would like to congratulate the Society for Alternative to Animal Experiment, SAE India, their office bearers, particularly Dr. Vyke Gupta, the President, and Dr. Amir Nazir, the General Secretary, for their tireless effort in making SAE India in full pace. So we have 3 speakers, 2 national speakers from Lucknow and Hyderabad, and 1 international speaker from USA. So the first speaker for this third session 3 is Dr. Amir Nazir from CSIR CDRI Lucknow. He has his education from the field of toxicology, obtaining his master's and doctorate degrees in the subject of toxicology, followed by a postdoctorate at Medical College Georgia, USA, and advanced research at the University of Nottingham, UK, and University of Freiburg, Germany. He is currently heading the laboratory for functional genomics and molecular science, where his research is employing on various functional genomics and epigenetics approaches towards understanding the mechanism of neurodegenerative disease. His research group holds the credit of identifying novel circular RNA molecules that are associated with protein quality control. His group aims at exploiting the protein clearance machinery and establishing the specific RNA signature as therapeutic model for neurodegenerative disease. So he uses actually C. elegans as one of the alternate model to understand the neurodegeneration. Dr. Nazir has numerous accomplishments to his credit. He has been awarded with the prestigious Raman Research Fellow by CSIR Government of India, and he is the Young Scientist Award by DST India, and he is also an elected Fellow of Society of Applied Biotechnology. The research work from his group has wide recognition with several publications. Now over to Dr. Amir Nazir to start his talk. Dr. Amir Nazir, welcome. Dr. Nazir, you are there?
Yes, am I audible?
Yeah. 15 minutes for you.
Yes, you are. Are my slides up there? Visible?
Yes, visible.
Yeah, visible. Slide is.
Thank you, Dr. Idris, for that very generous introduction. And first up, I would like to thank the organizers of this wonderful webinar for inviting me to deliver this talk. I would like to thank the President, Professor Vyke Gupta, Dr. Akbar Shah, Dr. Raisuddin, and the entire organizing team. So, in this short time that I have for this talk, what I will try to present would be just the application aspect, since many of the wonderful presentations did discuss the utility of the alternative systems as well as the regulatory aspects and so on. So, in my presentation, I will try to present to you how we employ the model system C. elegans in understanding the disease processes of humans. So within this domain, we have been trying to understand the mechanistic aspects of neurodegenerative diseases, Alzheimer's and Parkinson's, and we try to employ functional genomics approaches towards identifying what could be a common trigger for the multiple factors that are associated with these neurodegenerative diseases. So to that end, I would be presenting a small data set on identification of one of a very interesting microRNA molecules, which is called microRNA 4813, and which we believe could be a common trigger associated with multiple functions of these diseases. So the larger focus of our research laboratory is trying to understand neurodegenerative diseases and aging processes. And we employ this model for studying fundamental disease biology via identification of novel genetic modulators using functional genomics approaches, and towards understanding mechanistic aspects of aging and longevity in association with age-related diseases. We also employ this for drug discovery approaches, carrying out screening of new chemical entities, employing the transgenic strains that express human proteins. So that's what the advantage over here is, that these transgenic strains that we employ are expressing human proteins, which makes it easy for extrapolation of the data towards human conditions. And we also go on for further validation of identifying the modulators vis-a-vis associated functions within the worm model. So I will try to explain how. So just a brief overview of when we try to employ these model systems, because many a times when I'm defending grants, when we are presenting papers, we encounter a question that how come these small model systems can be relevant in studying human diseases. So there are some of the success stories which have come out of research with small model systems. So what we know is that, say if we talk about Drosophila, these are fruit flies and which are often called little people with wings. So why so is because most of the signaling pathways that define humans are conserved in these fruit flies as well. And functionally, when we speak, if we feed fruit fly caffeine, it has trouble falling asleep, something which you would see has a similar effect in humans as well. And feed it antihistamines, it cannot stay awake. Again, a similar physiological function as you would see in higher mammalian systems. And if we talk about worm C. elegans, a worm placed on the antidepressant fluoxetine has increased serotonin levels in its tiny brain, which would be similarly expected in higher mammalian models as well. And a unicellular model system, which is employed in genetics-related studies called yeast, if it's treated with chemotherapeutics, it stops their cell division, and removal of a single gene from mouse or zebrafish can cause animals to develop Alzheimer's disease or even heart disease. So there is, we're talking about zebrafish, there's a model where we can almost create like myocardial infarction-like condition because it has a completely pumping heart, and these models can very wonderfully be utilized towards understanding human disease conditions. So the C. elegans model, what it is, it's a small parasitic nematode, 1 millimeter long, 80 microns wide. So I will not go into the details in the interest of time. But what I would like to highlight in this slide is the importance of C. elegans system in studying neurobiology, because we know that when we talk about these wonderful genetic model systems, including Drosophila, yeast, zebrafish, C. elegans, each of these models have their own advantages of being employed whatever problem we are trying to study. For example, Drosophila having advantage in studying developmental biology and so on. In C. elegans, the essence is its nervous system, because when we want to understand a particular neurobiological function, or we want to understand a process or a disease condition, either we go to a very simple system, for example, in vitro systems, the cell lines. But if we try to study a drug, what we would be necessarily studying in the in vitro system would be the parent drug per se, because the whole organismal environment would be missing, the metabolism would not be there, so we will be studying the parent compound as is, but which is not the case when we administer such compound in humans or any other mammalian systems. So if we move from the simple in vitro system, what we next have is a complex mammalian system. So if we talk about the complexity of these systems, when we compare C. elegans, which has specifically 959 somatic cells and precisely 302 of these being neurons, that makes it very easy for studying these particular cells, because what we can also do is identify each of the neurons. We can even create cellular ablation models, thereby study which particular function a particular cell is actually undertaking. And if we compare this with that of humans, we know that humans have, like in case of C. elegans, the 302 neurons and 7,000 synapses, compared to human brain that contains about 100 billion neurons and probably 1,000 times more synapses. So what now the C. elegans system offers us is basically a very simple system which offers whole organismal environment with intact metabolism, and yet not offering such complexity as any mammalian model would offer. Hence, within this compact system, neurobiological processes, functions, and diseases also could be studied. So the development of these each cells is reproducible, and each of the cells, as I mentioned, have been identified, and the entire circuitry has been reconstructed. So of interest is this last point, that almost all gene families involved in neuron function in mammals are present in the worm as well. So employing these model systems, so what in our lab, what we try to do is we employ some of the very interesting transgenic strains. As I mentioned, they express human proteins. So talking about neurodegenerative diseases, be it Alzheimer's or Parkinson's, what we know is that the proteins that are responsible for these diseases, or at least are associated with these diseases, like amyloid beta in case of Alzheimer's disease and alpha-synuclein in case of Parkinson's disease. So what we know is that normal people also synthesize some amount of these proteins. So the difference between a normal person and a patient is that the clearance machinery in case of patients is defective, which means the proteins which are synthesized are not efficiently cleared by the cells. Thus, the proteins get accumulated and aggregated and causing toxic effects. So what we now know is that the protein clearance machinery, the protein quality control, overall quality control of the protein is actually affected in these patients. Now, what we do is that employing human protein in this transgenic strain, which is expressed within the worm model under the promoter of certain fluorescent proteins and other very specific markers, which actually led us in studying the endpoints related to these diseases, which I will not go into the details in interest of the time. But what it essentially helps us is towards carrying out very quick and high throughput screens of functions related to the neurobiology within these worm models. For example, the Alzheimer's related model that we have, if we expose in a normal condition, this model would have an increased aggregation, and that would ultimately, because the strain is designed in a way so that the worms have an increased expression driven inside the muscles, which leads to aggregation, thus causing paralysis within the worms. So if we are screening potential drugs and we see that in a high throughput manner, say if we are using even a 96 well approach, and we see that in some of the wells, the worms are not paralyzed, and then we would go back and try to see what was the drug that was given in that condition. And of course, we'll validate, we'll not end it there only. We would validate studying the protein condition, then in case of Western blotting also, and quantifying the aggregation versus soluble form of proteins, and then thereby screening that pharmacological agent, or it could be a genetic intervention in case of RNAi-induced silencing or any other such intervention that we are studying. So using these approaches, what we try to understand is the overall protein clearance machinery. And the strategy in this particular study that I'm presenting today, we carried out a global microRNA profiling. And the hypothesis behind that was, well, I would not go into the details of these methods in interest of time, but we essentially tried to study the downstream effects related to overall behavior, the dopamine content, the dopamine-related functions, because it's related to Parkinson. We also tried to study the cell death-related mechanism, including autophagy and apoptosis, and finally studying aging and longevity. And using the global microRNA profiling, the objective was actually to see whether we can identify, because we know that a single microRNA molecule can actually control or regulate the expression of sometimes hundreds of genes. So we started with a broader hypothesis that could there be a single microRNA trigger which could actually be controlling multiple functions, since we know that all these neurodegenerative diseases are multifactorial in nature. That's in fact one of the reasons that there is no complete therapy for these diseases as such, because alongside dopaminergic decline, there is dopaminergic cell death, there is like loss of these neurons, there is aggregation of proteins, there is reactive oxygen species, oxidative stress, and so on. So whether there could be a molecule which could actually be governing multiple genes that are related to these multiple factors. So towards this, we carried out global microRNA molecule, and out of lots of studies, which I will not go into the detail, I will just summarize it here. We identified two very interesting microRNA molecules. One of those which we took forward was this microRNA molecule 4813, and then we went on to study its downstream effector molecules. And among all of the genes, one of the molecules that was actually most relevant is a gene called VHE5, wherein which when we silenced using RNAi-induced gene silencing, we saw that the aggregation of alpha-synuclein was the highest in this, which means its absence was actually messing up aggregation very badly, which also means that its presence is required for effective clearance. And then we went on to identify other related molecules of these particular pathways, and we saw that we had 6 very interesting targets that were downstream effector molecules of 4813 that included genes that were related to apoptosis, degradation of lysosome, and ubiquitin-related processes, peptidases, and we had certain other molecules which were related to acetylcholinesterase and glutathione peroxidase, and so on. So we then went on to study related functions. For example, this strain, the picture that I have up here is from a strain that expresses green fluorescence protein specifically in 8 dopaminergic neurons. So that's another advantage of using C. elegans in neurobiology. It has identified neurons, as I mentioned, and we can tag specific neuronal subpopulations with fluorescent proteins. And in this case, 8 of the dopaminergic neurons are tagged with green fluorescence protein. We can use specific dopaminergic toxins to ablate these neurons and then see whether any of the conditions of genetic intervention or any pharmacological intervention can help us prevent or even reverse the condition of the dopaminergic damage. And here we identified that the downstream effector molecules of 4813 were really involved, particularly BHA5 that we identified in this. So with this slide, I would actually be concluding that out of the hundreds of downstream effector molecules related to 4813, we identified these 6 very interesting targets, BHA5 being the best one, which we are taking forward. And part of this study is already published in a journal called Disease Model and Mechanism, and we are taking the BHA5 story forward and trying to carry out studies related to electron microscopy, and where we are really trying to identify and pinpoint the dying dopaminergic neurons using electron microscopy in these worms. And we also want to study the mechanistic aspects, as this particular molecule happens to be a lysosomal VATPase having role in lysosomal acidity, and via that it is actually messing up the aggregation of amyloid beta as well as alpha-synuclein in the disease model. With this, I conclude. I hope I kept time, and I would like to thank my team and my collaborators and funders. And I once again thank the organizers for giving me this opportunity of presenting today, and I will be happy to take any questions. Thank you.
Yeah, excellent talk by Dr. Amir Nazir. We have a couple of questions we can take. Time for a couple of questions. Okay, last question. Dr. Amir Nazir.
Yes, sir.
Have you studied the same thing with zebrafish?
No, we don't use zebrafish, but yeah, there are like neurobiology associated models in zebrafish as well. But yeah, we work with the elegant system specifically.
Because it will be more interesting to study in a vertebrate model.
Right. So our larger goal usually is, I mean, we do not end it here. Once we identify something, for example, if we are carrying out a screening related to NCE, and if we have to take it forward, so what validation has to be done in the rodent model. So I collaborate, I mean, within our division, we have established what we call as 3-tier screening approach. And in that, what we do is, after I identify, because here C. elegans offers us an advantage of carrying out high throughput screening with like it's lot price efficient also and time efficient as well. And once we have identified certain targets, it could be a genetic target or it could be a pharmacological agent, we go for validation with other in vitro systems and as well as rodent models, which are very well established. So for Parkinson's, we have chemical models so far, and we are also in the process of procuring genetic models, including those expressing like mutated alpha-synuclein. So the validation obviously has to be done in this 3-tier screening approach before it can be taken further for mammalian use.
Very good, very good. Any other questions, please?
Can I ask a question, sir?
Sure, Dr. Pratisha, go ahead, please.
Yeah, good evening, Dr. Amir. It was indeed a beautiful presentation. I just wanted to know, do we have any validated evidences within patients who have known Parkinson's or neurodegenerative disorders having this miRNA expression levels?
Okay, no, since this is a novel kind of study that so this 4813, we are reporting for the first time. So
Okay.
yeah, that's a very interesting comment that you make. And our goal, larger goal, is actually to move on to validate it in human population as well. In fact, I have a collaboration going on with some of our clinician colleagues and collaborators at an institute here in Lucknow, RML, Institute of Medical Sciences. There we had proposed actually to carry out this microRNA profiling in human patients as well. So we would be, that's what our goal would be. We would try to correlate whether the ortholog of this particular microRNA, or in fact, we can do a global microRNA profiling there also and try to identify, because if at all we would be able to identify any of such microRNA molecules which are kind of associated with these diseases, in addition to being therapeutic of therapeutic relevance, they could also be used as diagnostic markers as well. I mean, though that's very ambitious, but yes, that is a direction to follow.
Yes, I absolutely agree with your secondary concept of diagnostics. But I just have one input. I think if we would have gone the other way around and seen a transcriptome analysis in patients and then gone back down to find out the mRNA was responsible for the transcriptome coming from the patients, and then modifying it in C. elegans to see whether it has any significant pathological effect, that would it have modulated the approach?
True. No, either way is fine. I mean, what we believe, either way is fine, because once we have these almost like humanized kind of models, any association here would potentially have an output over there also. And many a time, see, the disadvantage, and the challenge of studying human condition is that all access that we would have to in case of a patient would be circulatory fluids, right? So we'll get blood, we'll get CSF, and if we want a tissue, it would be only in a case of post-mortem samples. So that's the challenge, and that's where models like these come actually very handy, where we can study first and then we can try to correlate. In fact, there have been certain, so in the interest of time, I did not go into such details, but there are comparisons that have been carried out between these worm models, which are expressing human proteins, with that of the post-mortem human samples. And the proteomic profile has been shown to be similar. So those are some very interesting studies that have been carried out.
Because technically I think that most of the mRNA modifications may not get translated into pathological protein deformities, because generally what happens in post-translation modifications, most of the things get altered. So there is a possibility that even though you have an miRNA high expression level, it may not give you any pathological signal when it comes to the human data.
Very true. And that's why
I will intervene here. Dr. Deepti, you can discuss it, Amir.
Yeah, we could take this offline in the rest of time. Yes, that's
I'll get in touch with you, Dr. Amir. Thank you.
Thank you, Dr. Patricia. Thanks, Dr. Idris.
Thank you, Dr. Amir Nazir. It was indeed an excellent presentation. Thank you. The next speaker is Dr. Surat Parvatam from Hyderabad, from my own CCMB, Atal Incubation Center. Dr. Surat Parvatam did her PhD from TIFR Bombay the year 2016. She has been working as a science writer for Nature India, ScienceDevelopment.net, Wire, etc. She joined Center for Predictive Human Model System, CPHMS, at Atal Incubation Center in CCMB. CPHMS is a joint initiative of Human Society of India and AIC CCMB for the past 1 year. She has been working on various initiatives to enable human relevant research in India, including white papers, publication, workshop, and webinar series. She has recently organized a roundtable for academic, industry, and private funding bodies to initiate a dialogue and engage various stakeholders to address the current challenges and further advance in the field in India. So welcome, Dr. Surat Parvatam. She is going to talk about shifting towards human relevant and predictive paradigm for research and drug discovery in India. Go ahead, Dr. Surat Parvatam.
Thank you, Doctor, and thank you, organizers, for giving me the opportunity to speak in this platform. So I'll just try to share my screen. Sorry.
So,
Is it visible?
Yeah, you could able to share your presentation?
One second. So there are three options here, right? Present your entire screen, a window, or tab.
Entire screen. You please click on the entire screen and open your presentation.
Is it visible now? Is the screen visible now? Is the
Yes, it is. But not,
yes, it is.
But not the full. Yeah, now it is.
Yeah.
And
Yes,
perhaps you go for the full screen. Yes.
Okay. Is it fine now?
Yeah.
Share screen, then entire screen.
Ma'am. Slideshow, and then yeah, it is visible.
Yeah. Okay. So basically, I'm a senior research associate with the Center for Predictive Human Model Systems at Atal Incubation Center. And like it was mentioned in the introduction, it's a joint initiative between Atal Incubation Center and Humane Society International India. And this center was primarily established last year to enable human relevant research in India. And just as an introduction, biology, research in biology has been predominantly observational, where effects which have been observed in various processes. And these observations usually stem from animal models. And these observations which are observed during any process, they are then extrapolated to understand what would happen in human biology or in a specific biological phenomena. However, we as a center would like to enable a shift in this paradigm from observational to predictive science, where we move to a paradigm where we are able to predict biological consequences based on the vast amount of accumulated information that already exists and new information that we currently are beginning to generate from human relevant contexts and methodologies. So to do this, our primary focus has been three areas. So first has been how can we enable the generation of more data from human relevant contexts and methodologies such as organoids and organ-on-chip in India. The second focus has been, as we all know, currently there is no dearth of information. We have huge amounts of publications and data which is generated every year. However, there are very few frameworks or ways by which attempt to integrate all of that existing information. So few such frameworks have been proposed, such as the OECD Adverse Outcome Pathway frameworks, which then again attempt to integrate as to how some event at a molecular level causes something at a cellular level, which then triggers something at organ, and then finally an adverse outcome at the level of organism. So a few such frameworks have attempted to integrate this information to provide us with mechanistic insights into how any biological phenomena occurs. Interestingly, even in India, last year, the Department of Biotechnology initiated a program called the MANAV, which again, it is a crowdsourced citizen science program, which involves collecting the plethora of human biological data which is out there, and to kind of structure it to understand how changes at molecular and cellular level finally cause changes at organ or organism level. So such frameworks are beginning to be initiated in India and globally. So, but then our focus is how can we enable or further the use of these frameworks amongst scientists and researchers in India. And third aspect has been how do we then use this structured information in such frameworks, which can then help to feed on to computational models, which can then build predictive models of various native human biology or any specific disease model. So these have been the three focus areas. And again, I have put unidirectional arrows, but like biology, things don't happen unidirectionally. And all of these different focus areas are also interrelated to each other. For example, computational models or predictive models can also help in designing better human relevant methodologies or human relevant models. They can also help in designing better models like that. So all of these also cross-talk with each other, and they are not isolated areas. So to basically focus on these three areas are where we have been involved in various activities over the past 1.5 years. So these areas, these activities can involve, for example, writing white papers to understand what are the challenges to do research in these areas. And second has been, we have been organizing several training workshops, which help in training students and researchers to understand how to contribute to integrative frameworks such as adverse outcome pathways. We've also been, we also hold a monthly webinar series on various human relevant methodologies which are being done in India currently. Because one of the issues has been that many students and researchers aren't aware about these new methodologies. So we have a monthly webinar series where we have various researchers from academia and industry who come and talk about these methodologies. And we've also been involved in writing several publications and popular science articles to again increase more awareness and build on this knowledge base. So just like to briefly touch upon some of the insights that we found from the 3 white papers that we wrote. So we wrote 3 white papers, one on microphysiological systems, one on adverse outcome pathways, and third is on systems and computational biology and the challenges that are involved in doing these areas of research in India. So in terms of the microphysiological systems research in India, we found that it's a rapidly growing field in India where almost 25 to 30 labs have come up in this space in the last 3 years. And this research spans, for example, brain, liver, retinal, cancer organoids, and organ on chip. And this research has been focused on either developing these model systems to understand disease biology or for testing various candidate drug targets. In terms of systems and computation biology, which are kind of critical tools which can help to build predictive models of various human biology and also disease models. So we found that interestingly, almost 24% of all of published studies in India use in silico methodologies, which is very interesting and encouraging. However, most of these in silico methodologies are focused on certain areas of computational biology, such as sequence analysis. For example, they do not cover some of the other areas, such as model building or kind of building integrative models to understand how various different kinds of data, such as genomics or proteomics, how different kinds of data can be integrated. So some of the areas are less addressed, whereas it is much of the work is kind of focused on few areas of computational biology. And in terms of integrative frameworks, which are basically frameworks which can help in integrating existing information which is out there into structured formats. So one of the main aspects that stood out in our kind of analysis was that the general awareness for these frameworks is very low in India, that people are generally are not aware that such integrative frameworks exist and how they can basically help in identifying what are the gaps in current research and how such frameworks can also help in informing new experimental strategies. So this general awareness was kind of lacking both in students and researchers. So, and the main challenges that we found in conducting these areas of research is one, because this is such an emerging area, and because of its extremely interdisciplinary nature, the general lack of multidisciplinary and multi-stakeholder engagement is definitely an issue. For example, like we have listened to several talks on microfluidic systems, for example. So many of these systems also involve, for example, designing the flow parameters or designing the chip itself, and so which would involve, for example, collaboration with electronics engineers, and many of these microphysiological systems data also needs to be corroborated with clinical data, for example. So it also involves collaboration with clinicians. So that kind of multidisciplinary engagement is currently lacking. And secondly, many of the tools and reagents that are currently involved in to do these experiments is currently lacking in India, and most of them are kind of got from outside, which increases their cost several fold, which then becomes a significant factor. For example, if someone has to design a chip, there are very few microfabrication centers in India. Similarly, there is a general lack of government funding, and private funding is almost non-existent in this field. And also, if somebody, because as this is an emerging area, there are currently no training programs for early career researchers or students who want to get hands-on experience on how to build organoids or organ chips. For example, there are almost no or very few such training programs. And secondly, as in general, there are a lack of awareness that such methodologies now exist. So these are the main challenges that we found in this area. And just to briefly kind of touch upon the comparison of funding status of human relevant research globally and in India. So I'll just briefly touch upon in a few slides, considering we have limited time. So globally, these areas of both microphysiological systems and computational biology has been receiving a lot of attention, where for both these kinds of methodologies, there has been a significant funding. There have been significant funding initiatives. For example, there is a tissue chip program in US, and in EU also there are several such programs. And similarly, even for computational and systems biology, there is a significant amount of funding which is now been given globally. However, when we come to India, so we looked at how much funding is being given to these methodologies in India, and we primarily looked at the Department of Biotechnology as the major one of the funding agencies that we looked at, primarily because much of the data which DBT provides in terms of grants, it's all available and it's easily accessible. And secondly, DBT is the major funding player in the field when it comes to emerging funding of emerging technologies. So when we looked at the funding which has been given by DBT in stem cell or 3D methodologies, so as you can see, so dark blue is the total funding, and yellow is the funding for organoids and organ-on-chip projects. And as you can see, it's only a fraction of the total funding. And we also looked at number of projects which have been funded, because apart from the total amount, the number of projects also is an important parameter to look at. And as you can see, so the third column is the number of projects in organoids or organ-on-chip. So as you can see, in the last till 2013 to 2020, almost only 21 projects have been funded, and on an average, it's 1 to 3 projects every year. So it's still very less. And we also looked at funding in systems and computational biology, and it is marginally better than funding in organoids and organ-on-chip, for example. But it still needs it's still less than what we would want it to be. And and this is the government funding, and we also looked at private funding, and private funding is almost non-existent in any of these areas. So there is a definite need for both government and private funding to be increased in these areas. So to address many of these challenges that I've kind of spoken about right now, we also recently organized a roundtable with various stakeholders from academia, industry, and private funding bodies. And we discussed all of these challenges that we have kind of observed via writing the white papers, and also various interactions with scientists and people from industry. So we discussed all of these challenges, and several interesting recommendations were provided by various stakeholders. And and we plan to publish those recommendations that came out of that meeting. And hopefully, with this multi-pronged approach, where we are having both educational activities and activities for awareness, and then training and training activities, and also this kind of engagements where we have a multi-stakeholder meetings where we can now begin to initiate a dialogue between various players to understand where the challenges are and what do we need to do to address those challenges. So hopefully with all of these initiatives, we can begin to advance the entire field of human relevant research in India. So with this, I'll stop and I'm happy to take any questions. Thank you.
Thank you, Dr. Surat Parvatam. You can take a few questions if any questions are there.
You mentioned that there is a lack of private funding. So can you highlight what may be the reason behind? Are these concepts are not percolating well with the industry or private stakeholders? And what may be the factor which you observed for this?
Right. So this was also one of the main points of a recent roundtable discussion. And one of the main issues is that what some of the participants raised is that we need to first validate these technologies and establish the proof of concept. And so once we do that, so it kind of, and also have like a few success stories. And once we have that we kind of start building confidence amongst the investors that some of these methodologies are now validated and we have proof of concepts. And so we have like a few successes from these methodologies. So that kind of helps to build the investor confidence. So that has been, those were some of the points that were raised, that one of the main steps that we need to do is begin to have the necessary procedures to first validate and develop the proof of concept. So if we do that, then we can move on to the next steps. So
okay, thank you very much. Thank you.
Yeah.
So any more questions? So thank you, Dr. Surat Parvatam, for showing the other dimension of science. Excellent. It was an excellent presentation. Now I move upon to the third speaker of this third session, who is none else our international advisor for Society of Alternative Experiment Animals, Dr. Thomas Hartung. Dr. Thomas Hartung is from the United States of America. He has done his PhD MD, and he is the Dorenkamp Binden Chair for Evidence-Based Toxicology in the Department of Environmental Health and Engineering at Johns Hopkins Bloomberg School of Public Health, Baltimore. He has a joint appointment at the Whiting School of Engineering and also joint appointment for molecular microbiology and immunology at the Bloomberg School. He is adjunct affiliate professor at Georgetown University, Washington, D.C. In addition, he holds the joint appointment as professor for pharmacology and toxicology at the University of Konstanz, Germany. He is also the director of Centers for Alternative to Animal Testing for both universities. As a PI, he has headed a human toxome project funded by NIH, and he is the editor of Frontiers of Artificial Intelligence, and he has authored more than 580 scientific publications. Welcome, Dr. Thomas.
Yes, hello from Baltimore in the US. I would say good morning, you would say good evening. You can hear me well and see my presentation? Okay. Yes?
Yes. Okay. We'll accept it.
Wonderful. It is a privilege to talk to your society a second time this year. The last time I talked about COVID-19 and how it is a door opener for alternative methods. Today I'm going to talk about something completely different, which is the advent of artificial intelligence and big data in toxicology. It was very instructive to follow the talks so far. I was most grateful that Dr. Nat referred to our human toxome project, or Dr. Mohanan on pyrogenicity work we did earlier. So I really think this was a very interesting morning spent so far for me. You see me here with a mask, which we actually printed for our staff and board members, and it reads "saving more than animals." This is an important message for me. I'm a physician in a leading school of public health. My interest is to replace animals, but I do this in order to improve the situation of patients and consumers. I have to mention a few conflicts of interest or possible conflicts of interest. I'm going to talk about work we did with Underwriters Laboratories, UL, which is a safety standard organization and test service provider here in the US. I'm also sitting on the Apple's Green Chemistry Advisory Panel, where a lot of talk is about these methods. And I'm part of two companies. My former PhD student who did a lot of the work I will be showing, Tom Lichtefeld, has created ToxTrack and Insilica. So this is implementing some of this work. The other things are not relevant today. You already mentioned that I have another, let's say, interest. I don't see this as a conflict. I'm the chief field editor for Frontiers in Artificial Intelligence. And for Frontiers in Big Data and Artificial Intelligence, I'm heading the subsection for Medicine and Public Health, which is cross-listed. You see here our editorial board, some really renowned people, and we are very happy that this journal took off incredibly well. We have had about 200 articles, and we're expecting listing in all of relevant databases next year. Okay, toxicological approaches are helped by computers. I like to call this computer-aided toxicology. This was a review article in my Food for Thought series in 2009. And there's a lot of areas where I mention how computers are used in toxicology. But if you look a bit closer, AI or big data is completely missing. And this is only 2009, so about 10 years ago. How is this possible? Actually, artificial intelligence really was revived dramatically starting in 2012, and it is conquering all areas of our daily life at the moment, and toxicology is no different. Pharmacology is just the same. Suddenly, everything is machine learning, everything is AI. And the reason behind this is that AI is more or less an abbreviation for making big sense of big data. And big data sets is really a hallmark of life sciences in the last few years. To illustrate this a bit further, big data and AI are now fueled by a lot of different technologies. There is more and more databases which we are curating, which we are making public. There's the internet with its enormous growth and the data it is providing. There's literature, scientific literature. PubMed, as the most important sources, takes up about 2 million scientific reports, articles per year, and more than 80% of them describe substance effects. Not necessarily as toxicology or as pharmacology, but it describes how a synthesized substance is interacting with a test system. And then we have all these technologies, the sensors, the robotized testing, the various omics technologies, the high content imaging. Many of these data sources are terabytes per day. So it is really enormous what we are amassing here. In our own work, to give you a few illustrations, we were first of all interested in making sense of omics data. This is in essence what the Human Toxome project, which I had the privilege to head as a PI for the NIHS. So this was trying to interpret omics data better. And in my team, a lead role is with Alexandra Mertens to do this. So we were using technologies like weighted gene network correlation analysis, which we also translated to metabolomics. And Alexandra is continuing to work on making sense of large omics data set. This was a paper in 2008 where she analyzed the role of BPA. And I found it really remarkable after more than 12,000 papers on bisphenol A. She could very clearly show from these data it is not an estrogen receptor alpha agonist, but still many people assume it is just downstream merging with the effects of an alpha agonist. Or another paper from this year studying the relevance of cancer genes being studied from large omics data sets and patients. So this is one way of using AI and machine learning, interpreting data sets too big for us to comprehend as an individual. Another example for large data sets is coming from the exposome. People have not necessarily the same attention towards this development in the life sciences and toxicology, but what has happened with omics technologies to understand the activity, the dynamics of our substances is also happening at the moment with the imprint the exposure is making. I don't have the time to go into much detail, but it's a very similar application of trying to understand from the measurement of biofluids like blood and urine how exposures have actually impacted, and focusing then retroactively to identify what were the mechanisms and what were the sources which led to these changes in the human body. This summary article from a year ago is I think, a good starting point, and includes also as co-authors some of the opinion leaders in this field, which we did on occasion of initiating an exposome collaborative at Hopkins in 2019. The big work associated with our group was, however, something where we really tried to create predictive toxicology, where we wanted to make use of AI and big data in order to predict the toxic properties of untested substances. And to do so, we married two technologies which are well known, which is read across and QSAR. I don't think I need to explain in much detail, but read across is a very pragmatic approach where I look for similar chemicals and what I know about them and interpolate to my own. So very pragmatic, but it's very circumstantial, depends on what I happen to know. It's done manually, and it's very unclear when it is acceptable and when it's not. And QSAR is the classical approach of finding descriptors for molecules and using them with the computer to make predictions for substances which have not been tested. We combined these two, and we call this a RASAR, a read-across-based structure-activity relationship. So what the difference is here is we don't try to find one formula for the entire chemical universe, but for each and every substance, we just look into what are similar chemicals and what do we know about them. And the implementation of this 2 years ago did include 10 million chemicals, which meant more than 5 billion comparisons, one-on-one, were necessary. And more than 300,000 of them had biological information, 60,000 and more of them had animal data. This was already a very large database. And it took an Amazon cloud server 2 days to crunch these numbers with a 180-core computer to produce a map of the chemical universe, which is placing these 10 million structures in a way that we can see what is similar. So it looks somehow like this: a net where the distance between substances means they are similar. Then you can start entering information about their toxicity or non-toxicity and so on. We did use 74 different properties of these chemicals. Where we had information, we entered it. Where not, we left it out. And in total, 600,000 chemicals had data at this time. The work done by Tom Lichtefeld, whom I mentioned earlier, as part of his PhD originally, and then continuing with his company, was published in 2018. Within the first half year, the editor told me it was downloaded 24,000 times. It was covered in Science and Nature the same day we published it, because we showed in the same paper that 6 of the most relevant animal tests were on average 81% reproducible. But for 190,000 cases where we had a chemical with a known classification, we were 87% correct in predicting this. This was one of the largest cross-validations ever, 190,000 examples of known data used in this cross-validation. I don't have the time to go into any level of detail, but we have described our thinking, our development in a number of papers, starting with this one, making big sense from big data and toxicology by read-across. And then we continued working on this, on computational approaches to chemical hazard assessment. All of these are available in our journal Altex, open source, impact factor 6.2, by the way. So if you're interested to publish in this field, that's certainly important. A key element which really boosted the predictivity by about 10% on average was to use data fusion. And this means that if you look into eye irritation, we don't look what we know about neighboring substances on eye irritation, but we look also for, let's say, skin irritation. And something which is skin irritative is likely also eye irritative. We also look into mutagenicity because this is producing reactive chemistry, and reactive chemistry is more likely to be an eye irritant, and so on. So this is something which is making this enormously complex and data intensive, but at the same time it is teaching the computer what type of data are relevant. At the beginning of this year, a new version 2.0 was released. It now is based on deep learning, gives more reference to applicability domains, certainty of a prediction. It has potency prediction in as GHS classes as a non-validated part at the moment. The big news of this year was that the Australian legislation accepted this tool for 5 hazards. So since the 1st of July in Australia, you can use this also, including for cosmetic ingredients, to make predictions of human hazards. It is under evaluation by the FDA, and we are talking to other agencies, most recently to the European Food Safety Authority, about our tool. This is big. If you see the numbers, I only have good statistics for Europe, but the area which we might replace with the 9 tests it already has been validated for by cross-validation consumes 500,000 to 600,000 animals per year in Europe at costs of 200 to 300 million euro. So imagine, so much could possibly be replaced by this type of methodology if it stands a formal validation and gets acceptable. So this could really be a big bite out of the cake of animal testing. So I think this has a lot of opportunities which go beyond regulatory toxicology and formal validation and replacement. Just to hint you to green toxicology, the idea to use these type of tools early. Imagine a chemist could test already before he even synthesizes whether a substance has liabilities and is likely toxic. It makes no sense to further develop. This is an editorial which we had in Toxicological Sciences, which summarizes these type of ideas. So I would like to come to an end. Our vision at the moment is to make this even bigger. The last implementation from earlier this year already had 170 million structures. We added hundreds of thousands of data points. But we want to go further. We want to include also the knowledge from scientific publications, and our tool SysRef, and I say our, this is in silica and ToxTrack, which is a tool to help systematic reviews, is one of the bases for extracting information from the net. This is a tool free of charge to do systematic reviews with computer help. Another example is ChemChart, developed by the sister companies, which is mining the internet for safety data sheets and others. And we are in the process of bringing this into a European project of combining this as a data source. So let me close with Stefan Platz. Stefan Platz commented on our work at the British Society of Toxicology last year, and he said something which I really loved as a remark. He said, artificial intelligence, AI, will not replace toxicologists, but those who don't use AI will be replaced by those who do. So I'm most grateful for Stefan for this very pointed remark, and thank you for your interest in our work.
Thank you. Thank you, Dr. Thomas Hartung, for giving a glimpse on how big is big data and their view. So I think he can take. He'll be happy to take the questions.
Okay. This is Dr. Mani Vangala. Just very interesting presentation. Now, I just want to understand, do you think regulatory will accept that AI data that you're saying 9 toxicology tests can be reproduced with 80% accuracy? Would you think that regulatory will accept it, whether EPA or FDA, whatever?
First of all, I see this not as a tool to replace toxicologists, as Stefan says. Yeah, it is something which is, however, extremely helpful to decide whether a substance is likely to have making problems or not, and helping us to prioritize this. And with the expert judgment of an experienced toxicologist, you can take this and use it or not. I think it is clear already from our first implementation here that we are actually better than the reproducibility of the animal test, which is something which should really make us all think and in the end sing hallelujah that such a tool becomes available. But I would not recommend it as a standalone thing without expert judgment.
Okay, thank you. I think we need to talk offline. I have a lot of questions, and I think probably I'll give you a call sometime if I get your number. Please
contact me. I'm easy to find. I'm always happy to talk about this.
Sure.
So, very good. Data discrepancies, variation, artifacts, be it chemical or omics big data.
Yeah, this is one of the things we have to fight with. But for the good thing is that by using aggregate data, not looking only in one other chemical, one similar chemical, which most read across does, yeah, but looking into everything we know about all of the similar chemicals, you get an enormous push in quality because the artifacts are diluted out. The second thing is, I'm not advertising this as a solution for systemic toxicities, the highly complex things. Yeah, we are talking here really about the very basic toxicological properties, the topical and acute toxicities where this is working. I believe that we need much more biological similarity to understand how these things impact on reproductive toxicity, on chronic toxicities, and so on. Then we will not work with chemical structure only.
Yeah, that brings me to another question, actually, because back in 1995 to 2005, I was working at both Wyeth and J&J. We had a huge toxicogenomic program along with the Gene Logic at the time. And I was one of the people who actually reviewed most of the data. And it was a 10-year consortium program between 10 pharmaceutical companies and Gene Logic with chip that was available from rats at the time. Unfortunately, that data didn't really come any close to be able to predict it in terms of except for oxidative stress genes, we didn't see any specific biomarkers or toxicology markers in that data. So all the pharma companies stopped using that and they went back to regular chronic toxicity tests they do. It was about 300 to 400 toxic chemicals were used with 3 different doses, up to toxic doses. Do you have any of the data at your hand to be able to reanalyze it and say put into your big data format? Probably you should contact all these pharma companies who did it, and that would probably give you more depth in, because you don't need to do all these studies anymore. You can get that information from them and put it in there.
Yeah, it's this is easier said than done because the lawyers typically will not give you access to all of this because they say then the competition knows what we are working on or have been working on. And also liability is always an issue. But I agree that this is extremely valuable information because it moves us into the biological similarity of substances, which we need to move. However, in the early days of this, neither the cell cultures which were used or the animal tests which were used were sufficiently standardized. Secondly, the technologies have dramatically improved, the omics technologies. And at the time, they were not really analyzed with the tools of machine learning. We now have the computer power to really train on such mega data sets and identify patterns. We don't have to go in with, do I see a few genes which make sense? This is really a development which synergizes that we get standardized high quality culture, analytics, and the data analysis which is necessary. So I think it is all worthwhile reanalyzing.
Yeah.
Thank you. Thank you, Dr. Thomas Hartung. And again, I thank all the three speakers, Dr. Amir Nazir, Dr. this evening. And I, on behalf of SAE India, would like to thank Dr. R. Thirumurugan, Organizing Secretary, for organizing this event. Fantastic evening. Yeah, it's evening here, and for Thomas it is morning. So thank you so much, everyone.
Thank you.
Thank you. Thank you, Idris, for joining us and chairing this session. And thanks to Dr. Surat Parvatam. Who else in this session?
Amir Nazir.
Amir Nazir and Dr. Thomas Hartung. Thomas Hartung especially has been available with us and listening to all talks, the whole session. I'm so happy that he could join us and spend the time with us. And I have been always enjoying his approaches to alternatives. Whenever there is something huge talked about, he immediately adapts it from the alternative scenario. And this time, very interesting to hear from him about artificial intelligence and big data brought into the alternative scenario. Very interesting to listen to such topics from Dr. Hartung. Thank you, Thomas, for joining us. So we wrap up the technical sessions with this unit, and from now on for another 35 minutes we will have the rest of the proceedings. The annual combined executive committee and general body for 25 minutes, and 5 minutes for Dr. Vijay Pal Singh to sum up the proceedings, and another 5 minutes for Dr. Thirumurugan, the organizing secretary, to say thanks. So now we begin the annual EC and GB, and as Dr. V.K. Gupta, our president, will be the chairperson for this proceedings of this session, and he will begin with the session with an introductory note. Dr. Gupta. 5 minutes, please. Giving a caution note to the president of the society.
Thank you very much, Dr. Akbar Shah. And I would say that in spite of the technical glitches which a couple of our speakers faced, we are reasonably in time. And but I will like to cover up the overshoot of time by 1 or 2 minutes. And I would say, beginning of that my great appreciation again to Dr. Akbar Shah. He was not well for quite some time, and in spite of that, he has been able to knit so many excellent speakers. I think the top authorities in regulatory system and in toxicology area, in those who are working on alternate to animals in India, and knitting the international experts. And I am extremely thankful to my international experts that they have been able to, in such a short notice, delivered such an important talk. And I would recommend that maybe when we have next our annual conference, though this annual conference was a short duration of 3.5 hours instead of 2 days, but I think the impact of even this conference was not anyway less. There are two important things that we have been ensuring the regularity of the conference. This is the third conference, and in spite of COVID, we did it. And number two, the talks were so highly informative, and then the international people also participated. So this is something I think very, very, very good sign. And our membership has also gone up, though this is an area where there is a less of funding but in spite of that, we have been able to manage it. And so we should all make an effort that how we can percolate the culture of 3R or developing some alternative method where possible. And this culture should go not only to the bigger institution, but also to the universities, to colleges, wherever they are doing it. And then only people will think before you sacrifice an animal, why you're sacrificing and what maximum information one can derive from it. So these things are very crucial, important. And ultimately, we have to put our efforts to the government, to the policy makers, and also to the regulator. Policy makers means to the education department, and to rationalize the things, to the science and technology department to assist and fund it, and also to regulators so that they can also adopt the validated methods of alternate to animals. With this, I again thank the organizers, particularly the institute in Thiruvananthapuram who did all this logistics, and all my colleagues. And with that, I give it to Dr. Akbar Shah for delivering the annual report and take the proceedings on. Over.
Thank you, Dr. Gupta. I'll just bring up my presentation. Can you see the PowerPoint now?
Yes, we can see, Dr. Akbar Shah. We can see your presentation, Dr. Akbar Shah.
Yeah, I'm orienting that. Yeah, I come to the annual report, which should be as brief as possible. I get only 10 minutes to do that, and I share a part of the time with Pradiksha, whom I will introduce in the course of this presentation. So, Society for Alternatives to Animal Experiments, India, Annual Report 1920. Right? And so, we are in the midst of COVID-19 pandemic, which crippled the whole world. It also crippled the alternatives world. The World Congress, which was due to have been conducted in 2020, is postponed to 2021. Nobody knows for sure if it will be held in 2021 also. And there are other reputed conferences in the alternative scenario, like our American Society for Cellular and Computational Sciences, Biology, the European Society for In Vitro Sciences, had to postpone the meetings or go to the virtual mode, right? Okay. So we also had a problem, so we couldn't conduct the meeting as originally scheduled. Where we attend the meeting, so we have also taken to the alternative method. So and because of the COVID, there was not much of activity, but still I have some material to report. So the third conference assigned to the National Center for Alternate Animal Experiments, Bharatdasan University, could not be organized in the manner expected, yet thanks to the administration of the National Center, this virtual conference is in place. Let us thank NCAA, especially its coordinator, Dr. Ramasamy Thirumurugan, and his team, for faculty and students for the nice gesture.
Just 1 second, just one interruption. And before you go to the next slide, we would like to place on record our thanks, society's thanks and appreciation to your organization and to Dr. Ramasamy for excellent contribution, without which this could not be done. So So we place on record.
Recorded, sir, and it will be
In a very short time, very well done. Very well done.
Okay. Thank you, sir. We will convey it to Thirumurugan when he comes to talk at the end. And to report to you, the Joy Research Foundation, Vapi, Gujarat, and PETA India joined our society as industry and service organization members during 1920, and it has been formalized during this year. The society records its gratitude to JFR and PETA India. Dr. P. Srijit, Assistant Professor, Department of Zoology, University of Kerala, Trivandrum. Dr. A. Ranjini, Scientist, International Institute of Biotechnology and Toxicology, Chennai. Dr. B. Sabir Kumar, Assistant Professor, Department of Biochemistry, Central University of Kerala, Kasaragod. Dr. G. Madan, Associate Professor, Department of Biomedical Science, Bharatdasan University. Dr. Subramaniam Wangala, CEO, Reagene Biosciences Private Limited, Bangalore. Dr. Shikha Sarma, Postdoctoral Associate at Department of Environmental Medicine and Toxicology, University of Rochester, New York, USA. They have joined the society as life members. Let us welcome them to the SAAE family. Your General Secretary was invited to address the Japanese Society for Alternative Animal Experiments Annual Conference on the topic Non-Animal Methods of Drug Testing and Vaccine Development in India in the context of COVID-19 on November 13, 2020, and the Korean Society for Alternative Animal Experiments on the topic Alternatives Movement in India, the Past, Present and Future on December 18, 2020. Let us record our appreciation to these societies for the invitation to our society and the honor bestowed thereby. The Japanese Society is spearheading the formalization of the Asian Federation of Societies for Alternative Animal Experiments, which we discussed in the first presentation. And our society has already expressed consent to join the federation. Let us record our appreciation to the Japanese Society, especially Dr. Hajime Kojima, its former president, for the effort and endurance. Dr. Kojima has suggested that India can conduct our society can host the 2025 Asian Congress, and let us consider it in all its seriousness in the future meetings. The second annual conference of the society was conducted at National Facility for Biopharmaceuticals, Mumbai, on 13-14 December 2019 in a manner very grand. And the Episkin L'Oreal Travel Awards were disbursed at Mumbai itself, but the oral and postal presentation prizes are due to be conferred in the form of gold medals. The medals have been since made, but can't be handed over to the winners now in view of the virtual nature of the conference. I am painfully announcing that the scholars or students will have to wait perhaps for one more year to receive the medals in person. Some of them should be listening to today's proceedings, so I am making this announcement. An amount of 23,320 has been received from National Facility for Biopharmaceuticals, Mumbai, this being 10% of the registration charge due for the society. And let us place on record the contribution of Pradiksha Palage, the NFB and NFB team, and the principal management of the GN Khalsa College in Mumbai in organizing the conference and for the amount to the society. As per decision of the general body of the society, Dr. Pradiksha is due for a felicitation. The General Secretary shall send a certificate to that effect to Dr. Pradiksha after this virtual meet, because we can't physically felicitate her, though we are in distant places. The General Secretary places on record the great support from members, EC and fellow office bearers, the PETA India, the Humane Society, the Humane Society International India, for the wonderful support. So having given this brief report, I now invite Dr. Pradiksha to the in charge of the National Facility for Biopharmaceuticals to speak very briefly about the way she conducted the conference last year. Dr. Pradiksha will join us now.
Thank you, Dr. Akbar Shah. I will just share my presentation. So can you just exit presentation so I can share?
You can proceed now. Right. So that is Pradik Shah, in charge of the National Facility for Biopharmaceuticals at the Khalsa College in Mumbai.
Hi, Pradik Shah. We appreciate.
Hello, sir. How are you?
Fine. We appreciate your efforts.
Thank you. Thank you so much. I'm just unable to share my presentation. Yeah, good to go. Is my presentation visible?
It's come.
Yeah?
Yeah. You can make it full screen.
Yeah. So, good evening, everyone. Thank you for the time and patience. I am Dr. Pratiksha Pillai, Head, National Facility for Biopharmaceuticals, GN Khalsa College, Mumbai. I'm presenting you an overview of the NCAE 2019. The conference was organized by NFP GN Khalsa College, I being the organizing secretary. We had over 100 participants who took active participation in this conference. I thank the Society for Alternatives to Animal Experiments India, especially Dr. Akbar Shah, Dr. Raisuddin, and Dr. Phage Gupta for having the faith and trust in me and giving me this responsibility of conducting NCAE 2019. The conference was inaugurated by Dr. J.D. Yadav, who is the former VC ICT Mumbai, and we had several guests of honors, Dr. Smita Mahale, Dr. Rubina Ghosh, Dr. Kiran Mangav, Dr. Phage Gupta, and Dr. Akbar Shah, to name a few. The theme of the conference was Trends in Sciences of Alternatives, and the topics covered were in vitro approaches for toxicological evaluation, Indian scenario of alternatives, alternative model systems, newer alternative methods in context of education at different levels. We had the keynote address by Dr. Thomas Hartung. We had 16 guest lectures, 9 from international speakers, 7 from Indian speakers. We had 3 invited lectures. We had oral presentations by young researchers, which was 6 in number. We had poster presentations by 25 participants. There were 3 gold medalists in each of the category of oral presentation and poster presentation. And there were 2 travel grants awarded on behalf of the society. There was a panel discussion on regulatory aspects in alternatives to animal testing in India with all the stakeholders coming from the academic industry, regulatory, and others. The turn point event that happened in NCAE 2019 was the MOU between the Japanese Society for Alternatives to Animal Experiments, represented by Dr. Sakai, and the Society for Alternatives to Animal Experiments, represented by Dr. Vaidya. We had a pre-conference workshop on adverse outcome pathways, which was in collaboration with AIC, CCMB, HSI India, NFP, and Open Dogs. There was another pre-conference workshop at the Department of Zoology, University of Kerala. We also hosted post-conference workshops on handling of reconstructed human 3D tissue models, which was in collaboration with EpiSkin, the Society for Alternatives to Animal Experiments India, and NFP. Another of the post-conference workshop was held at Department of Elementology and Toxicology, Jamia Hamdard, New Delhi. The proceedings of the conference was presented at Altex, reputed journal, with an impact factor of 5.787. I express my sincere gratitude to the society, to the sponsors and collaborators who have generously contributed to the execution of this conference. My host institute, GN Khalsa College, the principal and the management, and obviously the team at NFP, without whom this conference would not have been successful. Thank you so much.
So thank you, Pratiksha, for this nice brief summary of the second national conference of the society. And we once again place on record your excellent contribution to the growth of the society and particularly making the presentation short and sweet today. Thank you. Now, having done this, I will now I request the chair to permit me to invite the treasurer to make the presentation of the annual accounts. He again will make it as brief as possible, allowing us to look through the PowerPoint, not read the details. So, Dr. Kadalmani, the treasurer.
Thank you, sir. Thank you.
Now, I'll exit again. Yes, you have come in. Right. Go ahead.
Please exit.
I think Pratiksha had to exit the PPT.
I have done that, sir. There is no presentation for my side.
Anyway, refresh the screen. Anyway.
So, good evening, everyone. So I have gathered here for this virtual conference for 2020. So I am happy to submit the financial statement of from 2019 to 2020. So this the financial statement, which is from January 2019 to March 2020. So the money is collected from those who are joined as a life member and the individuals and the industries and NGOs. The total amount is collected from the members, the 3,81,970. So these are the members who are joined in our society. So the total amount is the 3,81,000. So up to March 2020, so 3,81,970. So from that, so the expenditures is made strictly spent for the purpose of the society establishment. So initially we are the money is spent for the website and the registration, other things. And followed by the most of the money are spent for awards and honors. So right now, so up to March 2020, so the total expenditure is 1,88,891 rupees. The balance amount is 1,93,078 rupees. So here is the detail. The things is we will send through this WhatsApp for the further reading. So I request everyone, the EC members and GB members, I request kindly approve the financial statement for the auditing purpose for this the 2020.
Approved.
I'd like to make 3 comments on this. One is that the amount which has been provided by the NFB Mumbai is not accounted in this receipt because the money came during this financial year, and therefore that will be taken up or shown in the accounts for the next year. I think Diksha will be happy about to hear this. And the second comment is about the last year at Mumbai conference, we submitted a financial statement and got approval. But unfortunately, the auditor has objected to it because we didn't cover one full financial year, and therefore, the accounts that were submitted last year will have to be set aside, and these accounts will have to be submitted to the auditor for audit purpose and to be taken to the government to fulfill the requirements of the registration authorities. That's the second comment. The third comment is about the amount that was provided by the Episkin Laureate. It was roughly about 1.36 lakhs money that was sanctioned for the awards, the travel award and the award for the oral and postal presenters. And the money for the travel award has been already disbursed, and the medals have already been made, as I read it, and we will be able to hand over the medals only at the next meeting. So these are the three comments I wanted to present to the general body as a matter of alert. And if the general body approves of this annual statement, annual report, and the financial statement, it is approved.
Approved.
Okay, thank you so much. Now we move on to the.
Thank you.
Yes, sir. Yes, sir.
It's approved. Nice.
Thank you so much. So we will have 2-3 minutes to discuss matters that are brought up by the members. And if there are matters to discuss, which members would like to bring up, you're most welcome. I think this is the chairman's role to do this. Chairman, please take over. Discussion on the matters, newer matters coming up. Are there subject matters brought up by the members for this meeting?
I think we should include, try to make effort to include more members. I think not even 40. So I think there should be serious effort to increase the membership. Society is so far going very well, and new people, new faces are joining. So please, I only humbly request that those who have not yet joined, please join it and become the flag bearer of society. We need funds. We need. This time it was virtual, so not much money, almost no. But if we organize some conference in physical and in person, then maybe society has to also expend some money on success of that society. So with these words, I think I am encouraging my people at my university. There's some Deepti joined on my insistence. Many members are joining. So maybe we need to see. And some regular funding comes like Jai Research Foundation and this Fiskin, if other also come forward, and or like Dr. Mani can motivate some industry people to join. If he has been in the industry for such a long time. Some endowment fund, some kind of fellowship, travel grant to the young scholars can be created. It will be very wonderful. Some regular fund is to be given. As far as I know, government funding has become very squeezed nowadays. We are all facing. We got approval of research projects in month of January 2020. Still money is not coming. There are certain issues which we don't want to discuss at this forum, but some kind of industry support more to come forward. That is my suggestion, and maybe other members can add more comments. Yeah.
Thank you, Professor Reisdin, for the points made, and I request our senior members, particularly, as he rightly mentioned, Subramania Mangala, Suresh Posala, Idris, Amir, to help us, do a kind of lobby for us to increase our membership. More than money, respectability, the society comes through the number of members, life members we have.
And so, among students, they can popularize like Amir.
As a student member, yes.
I'm doing at my university. So at other institutions or research institutions or university, we should bring, and this concept is very nice, and we should. Yes.
So thank you. And I have a mail coming from Dr. Chandra Gowda Patil, who was formerly in Maharashtra. Now he has moved over to University in Delhi. He is now the admin of the WhatsApp group message ID of the society. He has just made a few remarks in his mail to present to this general body. The membership of SCAA India may include some perks like online training programs, free webinars, certificate courses, etc., which will attract more members, and the membership will mean something more than just a few conference events. I feel that SCAA should has some resource on its own, like a website to which only members have access. The second point, the members of the society should be provided with guidance, resource, discounts wherever they are trying to contribute to 3 years. This will attract real contributors. The society should develop regional facilities where members will be able to get training, discounts for testing. This is already there in the sense the Mahatma Gandhi Road and Camp Center, which I established, which has now become the National Center for Alternatives, is already doing it. And we are allowing for workshops and training programs, pre and post conference, every meeting. This time we are not doing it because it is virtual. Otherwise, we have been doing and giving a lot of training. We will continue to provide the training and subsidized facilities for doing the research. The society should conduct more focused events like the present one, so that people will be benefited more. There should be more online presence rather than face-to-face events. The actual in-person interaction should happen only when there are reasons. It is my frank opinion that conferences are becoming more like celebrations rather than scientific interactions. The virtual events have costs both for participants and organizers. So these are some of his observations, and we will take these points in the right spirit and consider the points in all seriousness in the years to come. And again, the point is that we should endeavor to enroll more members through all possibly positive approaches towards attracting people to enroll as the members of the society. So these are the comments. And are there any more comments coming from the members present? Dr. Gupta is around.
Hi, Dr. Akbar. This is Deepti. I just have one comment. Since we are hosting this event online and there's a lot of information and data, there are some restrictions on sharing data online with respect to third party. So I think we should also consider working on to guidelines on what can be uploaded online and shared online, and also take consent from all the participants before.
Yeah. That's an excellent point. I would agree that the thing is, especially coming from industry, our data is very proprietary, and we don't want any, because we don't want other companies to steal it, just to put it plain that way. And so we don't really share any data in most of these conferences. We talk about our concepts and what we'll do, whatnot, but we don't really want to share the data unless it is published or it's in public domain.
The points will really taken seriously.
Yeah.
And
Dr. Akbar Shah?
Yes.
I would like to say something.
Yes, please.
Yeah. So this is with respect to our previous discussions about the registrations to the society. I personally feel that if we do a small change in the administrative setup, like if we start having North chapter, South chapter, East chapter, and West chapter, and have 4 people in each of these divisions, North, South, East, and West, who will conduct routine activities, awareness seminars, or maybe, short lecture series within their zone to create awareness in the academic sector or in the research sector. This will help in creating awareness. It will increase the membership, and it will all happen under the umbrella of the society. So what is going to happen then? We don't have to travel logistically to various parts of the country to create awareness, but we can distribute responsibilities on zones and make sure that each one is working in each zone so that we can have better representation.
Plus,
I think the idea is good, but I think we should grow more. And then regional chapter will be more effective. We have 40 members, so some way we have to increase the membership. I think at least one chapter can be created either in your Bombay area or maybe in Trichy.
Your granddaughter?
Yeah.
I like Dr. Pratiksha's comment. This is Deepti, and I think this is in sync with Dr. Raisuddin, what we discussed when we had our meeting in before the first National Congress that was organized at Jamia Hamdard, and you were the organizing secretary. I think it definitely needs a lot more planning and active participation. But one point I would like to add here is if we can divide the responsibility of members, and then these members can collaborate with either universities or other research groups within their zone, north, south, west, or whatever, that would help reaching out more to other people who may not yet be very well acquainted with society and the work that we are doing. So that's just my impulse.
So I think I'll record a comment on this suggestion. We will seriously consider the points. And in fact, when I wrote the bylaws for the first time, you were not knowing about it. I was thinking of 5 regional centers: north, south, east, west, and central. Initially, I had it in the bylaws, but later I deleted for logistic reasons. Definitely, we will consider the point seriously, and we'll take one by one. We will introduce it as an aspect of the bylaws, as a recommendation of this meeting today, and then we will take it up one by one. We will start one in Delhi. Delhi will be a hotspot, and Mumbai would be another hotspot. Maybe we can start there, and then we will extend it to the other places. But Professor Raisuddin's point should be taken very seriously. At least 100 members should be a respectability to the society. Please do contribute. Each one can contribute the mite to increase the number of enrollment. As of now, points are taken. We will have the regional centers maybe in time to come. But in principle, it is agreed, and we will record it that way. Okay, Professor Raisuddin, you agree on this point? In principle, we agree.
Yes. One by one. We open one regional center, then next. Lucknow may be also one of the very important area.
Yes.
And Bombay and Trichy, or maybe we have to identify these places. And Kolkata, we have not touched much in eastern part of our country.
Yes.
Kolkata is very important hub of research. So we have to just identify some people and convince them. And yes, even these things will take shape, but whatever way we are going very nicely. And we made this particular today's event very nicely. And I think, and I am seeing that 64 persons joined at one point of time. So all 64 people, if they become members, we have ready-made 64 members. So yes. I request everybody, those who have not yet joined, please join us. And I think Dr. Akbar Shah, if somebody has problem, can we receive this live membership in 2 installments, if like that, if you agree? In some way we have to modify our bylaw.
In fact, please don't misunderstand me to be very preemptive. The point I have been thinking is that why not reduce the life membership?
Yeah, actually, I was thinking that
no, don't reduce it. Don't reduce it.
I think that you reduce it, you keep it same. But it was just laziness of some like me.
But at least in 2 installments, maybe.
2 installments. Okay, we will record it. We will record it and introduce it to the bylaws.
Sometimes research scholars and all, they are hesitant, but they want to join.
Yes. Yes, Professor Gupta. I think this is fine. 2 installments, we will record it and approve it.
To equal or some portion, 3,000,
3,000, 2,000. Like
okay, I think that's fine. That's fine.
Okay, fine. Thank you.
So that research scholars sometime they will join, and we can just tell them within 1 year.
Actually, I would do for the professionals. You should put maybe 10,000 rupees from industry, and academicians and everything 7,000, and for research scholars maybe 2,000, 3,000, students maybe 1,000, something like that. You should do that, so that way you can attract more people. But if you say all across 5,000 rupees for everyone, then a lot of students and research scholars won't join.
Or this installment system can be done for research PhD.
Even installment system doesn't work. I guarantee you on it. Okay.
Let us review it some more time, some other time maybe. Okay.
Yeah.
As of now, we will make it 2 installments, 3,000 and...
Installments work if they give a PDC.
Or I think within 1 year they have to give the next installment, something like that.
Yes, we'll record it and we'll include it in the bylaws.
And those members, they get some relaxation in the rebate in the registration fee, and it is the Patil raised quite important point that some free wear should be available. I think now this Confederation of Asian Society will be formed. Then definitely live members of our Indian society will get opportunity to get funding from, because Japanese are quite liberal in providing funding support. So I think I have been beneficiary. I am Society of Toxicology member. So I was giving travel fellowship to travel to Honolulu last time, and they gave me at least amount equivalent to my travel cost. So that the only members will be benefited. So and society will nominate them for such kind of travel award, travel grant. Then they will feel okay, this is an opportunity. And
so anybody, the question was asked that anybody who becomes the member of society will also automatically become member of federation, or they have to, so this was the question was asked. No?
I think that may not be possible because they have different membership criteria. I think only these kind of privileges members can get. Those who are Indian members, they can be only eligible for travel grant, conference support.
So for becoming member of federation, they have to apply again.
Yes, sir.
No.
Yeah,
it is actually each one is the member of the respective country society, and the country societies come together as the federation. So you have the privilege of becoming part of the federation activities, but federation doesn't have members. Only the national societies are the members of the federation.
Sometime federation may also charge money to become member. Like Professor Gupta, you know that for Society of Toxicology, we have to pay $3,000 every year to continue our membership of International Union of Toxicologists. Like that. So these are different scenarios we may consider. I think time is running short.
Okay, I got to go now. Dr. Gupta,
please.
I think Dr. Akbar Shafi asked Vijay to just say.
So last announcement from me would be that there is a proposal that the Kerala University Thiruvananthapuram would be interested in conducting the next conference. And I have told them that we will consider it in all seriousness in the days to come, taking opinion of other people who are also maybe interested in conducting the conference. So after getting all proposals, we will consolidate, and the executive through correspondence will decide on which will be the next venue. So is that okay? So Professor Gupta,
we fully agree with this, and we will visit next year Trivandrum.
Any institution which has at least 50 members should be given organization.
Reduce this number.
Maybe 10.
Okay. Yeah, they should bring up some members, right?
10 members.
So we will definitely consider it. And a few remarks from Professor Gupta, the president.
I think from
Vijay.
Big cheer, 3 cheers, and Happy New Year, and to all, and safe. And I think within 1 or 2 months we should be free of corona, and next meeting will be physical meeting.
Vaccine.
Let's hope for the best.
And Vijay is yet to speak.
I now invite Dr. Vijay Pal Singh, the Vice President, to present the concluding remarks. Dr. Vijay Pal Singh.
Thank you very much, sir.
Nice to see you on the screen.
Yeah. Thank you very much, sir. And I wish to thank the President, our vibrant President Vaike Gupta, sir, Raisuddin, sir, and also Dr. Akbar Shah, who are doing so much of efforts for the society. And I don't want to go much into detail each and everyone, but I found 2-3 things very interesting. As Dr. Gupta have said, 60 center we have on the Jabra phase, which is a very good point that now we have the relative alternative, not complete, then relative. Second, Gupta sir, I also touched about the large animal. But I am concerned about large animal, Gupta sir. I am sitting in so many ethics committee. We have done recently a systematic review. We are not doing what we are been supposed to do. I am not very happy. And where the experiment are being done, if you see the quality of experiment, like experimental design, sample size calculation, randomization, blinding, conflict of interest, statistics before and after, and we published this paper into ETLA very recently. So I think that is not the part of this, but not very good this one. Another thing which was pointed out by Thomas, that we have 9 tests, and for that we are using nearly 5 to 6 lakhs of animal. So probably we can choose something out of that 9 tests, what is saying out of this paper, and can focus on few, will be very good idea. Because I personally believe for typhoid fever, the vaccine took 100 years. For Ebola, it took 2 to 3 years. And for SARS-CoV, it took less than 1 year. So if need arises, if we keep everything open, if we take things positively, it is possible. So with these words, I don't want to take much of your time, and I am not contributing much nowadays because of my preoccupation with FSSAI. But as soon as I am free from there, I will 100% full throttle give my support for the society, and even from there. And with these words, I would like to thanks for the opportunity. And I see both of Professor Akbar Shah and Professor Vaike Gupta as my inspiration to continue work, which is with the no-nonsense attitude and straight way. Go ahead. Thank you very much, sir.
Thank you, Vijay, for your wonderful words. Now I'll call upon Dr. Tirumurugan, the coordinator of the National Center for Alternative Animal Experiments, the organizing secretary of this program, to propose the vote of thanks. 5 minutes. Dr. Tirumurugan.
So very good evening, and also say very good night to all. It is my privilege to have been asked to propose a vote of thanks on this international webinar and 3rd National Conference of the Society for Alternative to Animal Experiment. First and foremost, I thank Honorable President Dr. Vaike Gupta, sir, and Vice President Dr. Raisuddin, sir, and Dr. Vijay Pal Singh, and Treasurer Dr. Kadalmani, and our most valued invited speakers and participants from various institutions. My special thanks to General Secretary Professor Amir Nazir, sir, for his tireless effort for his webinar and his contribution to take this event to a height of the victory. On behalf of National Center for Alternative Animal Science, Bharathasan University, and other members, I thank you very much, sir. I really salute you, sir, for your efforts. We thank all the invited speakers, Dr. Hozima and Dr. Nath and Dr. Vangala and Dr. Deepti Kapoor and Dr. Mohan and Dr. Nazir and Dr. Surat Barbadam and Dr. Damas Hatum. And really much thankful to the chairpersons, so Dr. Raisuddin sir and Dr. Adip Rai and Dr. Mohammad Idris, who made this event very successful, and the delegates for the enthusiastic participation in this webinar. I express my sincere thanks to our Honorable Vice Chancellor, Professor Manishankar sir, respected our Registrar, Professor Gopinath, for permitting us and providing administrative support. I should mention our collaborators, Professor Arjunan, Professor Babu Rajendran, Professor Tamari Selvi, Dr. Achiraman, and Professor Tajuddin, Professor Nadaraj Srinivasan, Dr. Dhanasekaran, and Professor Parthasarathy and Professor Jagannathan for their valuable contributions and enormous cooperation in organizing this event. I am sure that this webinar contributed the new dimension of animal alternatives for scientific experiments, which helped the participants to gather the numerous information in the field of vaccine testing. The event would not have been successful without the volunteer of our research scholars in the National Center for Alternative Animal Experiments and the students in the organizing committee. I thank you so much for their valuable and wonderful efforts for successful of the conference and behind of our Professor Akbar sir. I thank everyone whose contribution has made this webinar a successful one. I also thank the University Grant Division and Mr. R.K. Raj, Senior Programmer and Director, University Informatics Center, Bharatdasan University for the support. Almost time is 10:30. And in the future, the society is planning to organizing more number of conferences and seminars towards the betterment of the non-animal models. We are the forerunners for greater alternatives to animal experiments. Finally, I honest to say all the credits goes to Professor Akbar sir and Professor O.K. Gupta sir and also Dr. Professor Raisuddin. I wish you all the best for your future career, and I am wishing you all an advance happy and prosperous New Year's. Very good night to each and every one of you. Thank you so much for this opportunity providing the propose the vote of thanks. Really a heartfelt thanks to each and every one of you, sir. Thank you.
Thank you, Dr. Thirumurugan.
So once again, on behalf of this society, let me thank Dr. Thirumurugan and his colleagues and his volunteers.
And we will visit your place. Don't forget. Yeah, and you have to host us. We will be your guests, definitely, sometime. In very near future, vaccine is around, so hopefully we will be there.
Chairman, please wrap up. One sentence.
Sir.
President.
Gupta, sir.
Please wrap up.
Mic. Your mic.
You have to unmute the mic. Dr. Gupta, please unmute.
Cheers to everybody and wish you all Happy New Year.
Happy New Year to you. Okay.
See you.
Yeah.
Let us hope to see each other in person next time.
All of us now at the last moment should say like this.
Yeah.
To Dr. Akbar sir, to entire team.
Thank you.
Thank you, sir. Thank you.
Thank you, sir. Call it a day. Thank you so much. I will exit. Thank you, Professor Michael, for joining us and all other invited participants. Thank you so much.
Thank you.
Bye.
Thank you, Dr. Everyone.
Thank
I wish you a very happy New Year.
Bye. Happy New Year to all. Bye. Bye.